Okay, so welcome everyone to the 56th lecture of Dr. Hydri Step 1. Before we begin, can we assure that you guys can hear my voice? Is my voice clear? Can you guys hear my voice? If you can, please use the chat box to let me know that you guys can hear my voice. Okay, very well. Thank you so much for attending the lecture. Hope you guys are doing well. First and foremost, let me assure if you guys did the questions yesterday or not. And if you did, how many of the questions did you guys do? That's question number one. Question number two is, out of those questions, how many did you get right? What was your percentage score? And the second and the third thing I want to know is, did the lecture help you guys out in solving the UWorld questions? Would anyone be kind enough to answer these questions? How many questions did you guys do? What was your score? And was the lecture helpful? Meaning, was it in sync with the questions that you guys received? Okay, Dr. Hassam, 80 questions, 60 right. Very good. 60 out of 80 is a very good score. Anyone else? 60 out of 80 is a very good score. Anyone else? How many questions did you guys do? And what was your score? Would anyone else be kind enough to answer the questions? How many questions did you guys do yesterday? If you guys do not give me an answer, then I would assume that no one did any questions. Anyone? How many questions did you guys do? 60, thank you so much. Out of those 60, how many did you get right? 60 questions, that's very impressive. 44 out of 60, that is very impressive. Thank you so much for putting your attention. And third question is, did the lecture coincide with the questions? Meaning, was it in sync with the questions which you received? Okay, thank you so much, good. Very good, because I always want a feedback before I begin the lecture so that I understand that the questions which you guys are doing nowadays, those are relatively new questions. You are welcome. The questions which you guys are doing nowadays are relatively new questions, so I just want to make sure if our lectures are in sync with the questions you are receiving or not. Anyone else care to answer how many questions they did and the percentages of questions they got right? After that, we shall begin our revision and recapitulation from yesterday's topic. Dr. Iman with 40 questions. That is very good. Out of those 40 questions, how many questions did you get right? $34 out of 40. That is very impressive. Thank you so much. Were those questions all related to genetics or? Very good. Very good. If you got 34 questions out of 40 questions in genetics, I know that genetics has only 94 to 99 questions, then you are doing absolutely amazing. So thank you so much for putting your attention in the lectures and in the UWorld questions. Thank you. Next is Dr. Hussein with 30 questions. How many did you get right out of those 30? $25 out of 30. These are amazing numbers. Thank you so much. That is absolutely amazing. Out of those 30 questions, were they all from genetics? Okay, that is very good to know. I'm so proud of you guys. Thank you so much for getting these numbers and getting the score. And okay, so with that being said, I'm not going to ask you guys anymore about the questions. Hope you all are receiving questions which are in sync with our lectures. If you guys have any feedbacks for us, please let us know. And with that being said, can we begin our revision and recapitulation from yesterday's topic, please, so that we can get ahead on our schedule on first day today? Okay. Is everyone ready with the revision and recapitulations of yesterday's topic? Okay. All right. Okay, so let's begin. First of all, if I talk about blood groups, HLA, and what type of genetic term would come to your mind? Codominance. Very good, Dr. Karbasi, Dr. Nikki, Dr. Evelyn, Dr. Uday, Dr. Hussein. Good answer. Codominance. Next one is neurofibromatosis. What type of genetic term should come to your mind? Variable expressivity. Okay, very good. BRCA1 and ovarian carcinoma. Very good. Phenylketonuria. Phenylketonuria. Very good. Huntington's disease. Huntington's disease. Anticipation. Very good. How about P53 mutation? P53 mutations. Nope. Okay, it's dominant negative. Dominant negative. How about Down syndrome? Mosaicism. Very good. Okay. If the mosaicism is present in a child, but it's absent in the parents, what type of mosaicism is this? Gonadal mosaicism. Very good. Okay. Beta thalassemia. What type of genetic term would you use to describe beta thalassemia? Very good. What type of genetic term would you use to describe Leber's hereditary optic neuropathy? Okay, good. If p squared plus 2pq plus q squared equals to 1, then what do we represent with p squared? Healthy allele. What do we represent with q squared? Okay. What do we represent with 2pq? Okay, good. Very good. Next question is, we have two types of structures which are responsible for degradation of mRNA. One of them is naturally produced, another one of them is produced in vitro. Which one of them is produced naturally for mRNA degradation that will stop transcription? MicroRNA. Very good. Which one of them is produced in vitro? Very good. siRNA. Okay. Okay. If I ask you that you have to perform a laboratory procedure in order to detect chromosomal translocation, which lab procedure would you use? Very good. If you have to detect chromosomal abnormalities or sex chromosomal disorders, which procedure would you use? Very good. Okay. If you have to detect hematological malignancies, which procedure would you use? Okay. If you have to detect cellular size granularity, which procedure would you use? Okay. If you have to perform DNA amplification, which procedure would you use? If you have to detect mRNA, which procedure would you use? Okay, good. Are we all clear about these things? Yes or no? Okay, good. So without further ado, let's begin with today's lecture. No? Okay. All right. So first and foremost, thank you so much for answering these questions with absolute perfection. From what it sounds like, first of all, do we have anyone who's looking at the book and giving out the answers? Okay, I trust you guys. Okay, I trust you guys, and I'm sure you guys are not looking at the book and giving the answers. If you are, that's still okay. Okay, meaning that revision is still a revision, but the best way to revise is not looking at the book and testing yourself. Okay, testing yourself. No time to look to book. Exactly. That's why I try to ask you the questions as fast as possible. And I give you guys a countdown. From next time onwards, I'm going to give you guys a countdown from 3 to 1. After asking a question, I'll ask you guys 3, 2, 1, and only 3 seconds for your answers. Okay, so no time to look. Very good. Okay, so that's that. And so we studied a lot of genetic terms yesterday. Herdy-Weinberg's principles, lab procedures. Then we did questions. Then we talked about USMLE preparations. Okay. From today, what we are going to do is we're going to see if we can make a dent out of the rest of the genetics that's here. And this is the first time today that we would be actively using physio. Did we use physio before? Did we use physio before? We did, didn't we? For the embryology? Yes, for the arches. Am I correct? For the first, second, third, fourth arches? Okay. So today we are going to use physio once again. And we have a lot of picture mnemonics in physio, which will actually help us out in remembering all the diseases. For example, we will use, is it possible to use your physio? Our physio, so the thing about the physio is, we do not want to give you guys the physio ID and password. I'm going to tell you why, okay? Because when you start watching physio, it's another resource entirely by itself. The last thing you need is to add another resource to your study schedule. So what we are going to do is we are going to select the topics from physio, which is absolutely important, and we will do that during the lecture. So what that would do is that would allow you to understand first aid completely, which is our goal. So instead of giving you guys the ID and password, which we thought we would, we decided that we are going to share the pictures, the mnemonics and everything during our lecture. So what that would allow us to do is make sure that you guys are not overburdened by a lot of resources. So that's what we're going to do. And you have absolutely nothing to be worried about because since we are going to use physio, we will make sure that we use physio to its maximum potential. Okay, so with that being said, can we begin the lecture? Yes or no? Is everyone ready? Okay, let me share my screen and let's begin. Okay. And here you guys are. Okay. Okay, so we did the Herdy-Weinberg principle yesterday. We are going to talk about uniparental disomy and imprinting. Okay, so let's begin with uniparental disomy. What is uniparental disomy? Uniparental disomy, the word itself, it means that offspring will receive 2 copies of a chromosome from one parent and no copies from the other parent. Uniparental, meaning that they will receive 2 chromosomes from either the mother or the father, and they will not receive any copies from another one. Heterodisomy indicates a meiosis 1 error, and isodisomy indicates a meiosis 2 error, or post-zygotic chromosomal duplication of one of a pair of chromosomes and loss of the other of the original pair. So you have 2 types of disomy. Heterodisomy, which is a meiosis 1, okay? Hetero is meiosis 1, and iso, 2 I's in iso is a meiosis 2 error. Okay. And what happens is over here, you have the knockout of either of the parent's gene. And after a post-zygotic chromosomal duplication, one pair of the chromosome, you have the duplication of one chromosome and you have loss of the other pair. The uniparental is euploid, euploid meaning correct number of chromosomes. Most occurrences of uniparental disomy will result in a normal phenotype. We will consider isodisomy in an individual manifesting a recessive disorder when only one parent is a carrier. So this knowledge will get fortified when we study imprinting disease. But basically, if you understand uniparental disomy, that's basically meaning that if you have the mom's chromosome and then you have the dad's chromosome, right, over here, every chromosome will make 2 copies, and the zygote would, for example, if a zygote receives 2 copies of the mother and 0 copies from the father, then this is a type of uniparental disomy. And uniparental disomy is euploid. This means that there's correct number of chromosomes. Most occurrences of uniparental disomy will result in a normal phenotype. So what would happen is the phenotype will be okay, it will be normal. And consider isodisomy in an individual manifesting a recessive disorder when only one parent is a carrier. So what happens is disomy can be of 2 types, that is hetero and isodisomy. Isodisomy is considered when the individual has a recessive disorder, for example, cystic fibrosis or any other types of recessive disease, and only one parent is the carrier. Out of them, if one of them is a carrier, then what happens is, if I tell you that you have an autosomal recessive disease, right? For an autosomal recessive disease to happen, do you need both parents to be the carriers, yes or no? Do you need both parents to be the carriers, yes or no? Fast answers, please. Okay, so over here, both of them have to be the carrier, right? But what if I tell you, okay, what if I tell you, one second. Okay, what if I tell you that what if I tell you that in a patient, if you have only one parent who is a carrier, in a patient, if you have only one parent who is a carrier, then if it's not a disomy, is it possible for the child to have the disease? Yes or no? If there is no disomy or isodisomy or heterodisomy, is it possible for the child to have the disease? No, it's not possible. It's only possible when both the chromosomes are coming from one parent. So for example, if we have, let's say the mother is a carrier and the father is not a carrier, in 100%, there's a 100% chance that the child will not have the disease if there's no disomy. If there is a disomy, meaning that the child receives all the chromosomes from the mother, but does not receive any chromosome from the father, then that would mean that this is a case of isodisomy, meaning that the 2 chromosomes are coming from the parent who is a carrier. As simple as that. That is your disomy. Is everyone clear about this? Yes or no? Is everyone clear about this? Okay, good. Very good. Okay, there are certain examples, for example, Prader-Willi and Angelman Syndrome. Okay, so what we would do is we would talk about imprinting first. Okay, please repeat it. Okay, good. No problem. Okay, please put your attention to this one. This is a very important topic which we're discussing right now.
Somebody wait.
Oh, someone's waiting. Okay, let me see. Someone's waiting. Okay. Nope, no one's waiting.
Okay, thank you.
Okay, no one's waiting over here. Okay. Okay. Can you guys see my screen? Yes or no? Can you guys see my screen over here? Okay, good. Okay, so my question to you guys is, if, let's say the mom, okay, this is the mom has A, the dad has A. Is it possible for a child to have an autosomal recessive disease if there is no disomy? Fast answers, please. No. Okay. What if I tell you that only the mother's chromosome are going to the child, and the father's chromosomes are not going to the child? then is it possible for the child to have the disomy then? I mean, the autosomal recessive disease then? Yes? Yes. How is it no? I'm going to tell you why. Because over here, if the mom is a carrier, then both of the chromosomes are coming from the mother. This is one chromosome, this is another chromosome, and then the child has a 25% chance of being a autosomal recessive diseased individual. Do you understand what I mean? Okay. Okay, so this is disomy. So the next time is, how do you recognize disomy? How do you recognize disomy? How do you recognize? No, he will not be AA. Okay, let me clear your confusion. This is one chromosome from the mother. This is another chromosome from the mother. Do you understand what I mean? Instead of the father, both of the chromosomes are coming from the mother. So the child will have either one of these chromosomes. So there's a 25% chance that this child will have the disease. Okay, so that is how children whose parents are a single carrier can have an autosomal recessive disease if there is disomy. If there is disomy. Are you guys clear? Yes or no? Is everyone clear about this? Okay. Okay. Let's talk about imprinting. Let's talk about imprinting. Imprinting is basically one gene copy is silenced by methylation and the other copy is expressed. Okay. Do you guys remember how we talked about the fact that methylation is makes the DNA mute in our first lectures? So if you methylate one copy of the gene, then there's silence of the gene. If the gene is silenced, do you think that they will produce the phenotype? Yes or no? No. So as a result, what happens is if one copy is silenced and the other copy is expressed, then this is known as imprinting. When we talk about imprinting, there are two diseases which we have to consider, and these are extremely high yield. These are extremely high yield. First of all, we are going to talk about Prader-Willi syndrome. Prader-Willi syndrome. So what's happening over here, that in Prader-Willi syndrome and Angelman syndrome, you have genes which are coming from both the mom and the dad, but one of them is being silenced. So over here, one of them is being silenced by methylation. That's what you have to keep in mind. Prader-Willi syndrome, which, if you ask yourself which gene is silent, the maternally derived genes are silent over here. And the disease occurs when the paternal allele is deleted or mutated. So what happens is, you have two chromosomes, right? Both of them are coming from the mother, and another one is coming from the father. What if I were to tell you that the chromosome which is coming from the mother, it's silenced, and on top of that, the chromosome which is coming from the father, that is deleted? Will that create a problem? Yes or no? Fast answers, please. Yes. And in the same way, if I tell you that you have a child whose chromosomes are coming from the mother and the father, and the chromosome that is coming from the father is silenced, and the chromosome which is coming from the mother is deleted, will that create some issues or troubles in the child? Yes or no? Okay, if that's what you say, then that's what it is, because that's exactly what's happening over here. And you will be tested on this time and time and time again, that which one is silenced and which one is deleted. And the best way to remember this is P in Prader-Willi. P for paternal allele is deleted, and M in Angelman to remember that the maternal allele is deleted. So if you come across P, that is Prader-Willi syndrome, if I tell you that the paternal gene is deleted, and then you can instantly identify that if the maternal allele is silenced, then this disease will happen. And vice versa, it goes to Angelman syndrome when there is deletion of the maternal gene and the paternal gene is silenced. Okay. Another question you will obviously receive over here is, how do you silence the genes? And the answer is by methylation. That's another one. And the third question which you will receive from over here is, what is the name of the paternal gene that is silenced in Angelman syndrome? The name of the paternal gene is UBE3A, right? UBE3. A. Okay, so this is the paternal gene that is silenced over here. Okay, let's look at some of the sign symptoms. The sign symptoms are for Prader-Willi. Okay, so when we think about Prader-Willi syndrome, we think about hyperphagia, obesity, intellectual disability, hypogonadism, and hypotonia. So what's happening over here is, if we talk about Prader-Willi syndrome, okay, if we talk about Prader-Willi syndrome, then we are talking about a very sweet little boy who is a bit on the obese side and not so much on the brighter side of intelligence. Okay, so Prader-Willi syndrome. Prader-Willi syndrome is this fat baby over here. Okay, okay, Prader-Willi. Okay, what are the clinical features? First of all, fat baby, so obesity. Why is he or she obese? Because of hyperphagia, meaning they have a tendency of eating a lot. Hyperphagia. Okay. Along with this, do you think if do you think a person who for example, in this child, what happens is, since there's obesity and hyperphagia, this child also has a problem with maintaining his tones, I mean his muscular tone. So he also has hypotonia. Hypotonia. Okay. Another one is he has intellectual disability. Intellectual disability. Okay. Intellectual disability. And another one is, if I ask you which chromosome is affected in both Prader-Willi and Angelman syndrome, the chromosome number is chromosome number 15. Chromosome number 15, and especially on the Prader-Willi, it's chromosome number 15 on the paternal gene. On the paternal gene. And in the same way, we have another sweet little boy over here. Okay, the disease is known as Angelman syndrome, and the word we're going to focus on is angel. Okay, Angelman. Angelman for a happy baby, meaning that the baby is always happy. Okay, so it's comparatively, it's a very happy baby. So there is inappropriate laughter. Inappropriate laughter, meaning that the baby is always really happy, as a matter of fact, which is happy to the point that it's actually inappropriate. Basically, no matter what you say to the baby will always keep on laughing. So inappropriate laughter, that's number one. Then two things for the CNS, that is seizure and ataxia. That is, the baby will have seizure and an ataxic gait. And another one is the baby will also have intellectual disability. The baby will also have intellectual disability over here. Okay. And once again, the chromosome number for this one is also chromosome number 15. And obviously the gene which we talked about was UBE3A, right? And the copy is on the maternal gene. Okay. So once again, Prader-Willi syndrome, which gene is deleted and which gene is silenced? Fast answers, please. Prader-Willi. Which gene is mutated and which gene is silenced? Can I get some fast answers, please? Okay. And Angelman syndrome, which gene is mutated and which gene is silenced? Okay. Okay, very good. So that's exactly what it is. And another thing is if you have, let's say, uniparental disomy, okay? If I tell you that you have uniparental disomy, okay, when we talk about uniparental disomy, do we mean that both copies are coming from one single parent, yes or no? Okay. What if I tell you that for example, you have a patient who is a patient of uniparental disomy, meaning that both copies of the genes are coming from the mother, but no copy is coming from the father, and the copies of the genes which are coming from the mother is silenced. Okay, hear this question out. You have a patient with uniparental disomy where both copies of the gene is coming from the mother. The copies of the genes which are coming from the father is obviously not coming because it's a uniparental disomy. And the genes which came from the mother is silenced or methylated. What syndrome would develop in that patient? Very good. Meaning, if there is maternal silencing, then it will be Prader-Willi syndrome. So we said that both copies of genes are coming from the mother, and the copies of the genes did not come from the father. As a result, the father's gene could be is absent, and the maternal gene is silenced. So the disease is Prader-Willi. The disease is Prader-Willi, and vice versa. If we talk about the fact that if you have a patient who received two copies of the genes from the father and no copies from the mother, and the paternal gene is silenced, so what is the diagnosis? Angelman syndrome. Okay. So can we conclude the fact that Angelman syndrome and Prader-Willi syndrome can occur due to imprinting or disomy? Yes or no? Okay. Okay. What would you call a patient who has an autosomal recessive disease, but only a single parent is a carrier? If there's a single parent who is a carrier, but the patient has an autosomal recessive disease, disomy, is it a heterodisomy or an isodisomy? It's a isodisomy. Very good. Very good. It's a isodisomy. Okay. So that's that. Okay. If no gene is coming from the father, only from the mother, how can it be Prader-Willi? What happens in Prader-Willi? In Prader-Willi syndrome, what happens in Prader-Willi syndrome? In Prader-Willi syndrome, you have the paternal gene, which is deleted, and the maternal gene is silenced. So in the same way, in disomy, if you have both genes coming from the mother, right? And those both genes are silenced. And obviously in disomy, the paternal genes are not coming. So they could be considered as deleted or absent. And the child will get Prader-Willi syndrome. That's how, okay, that's how we could do this. Another one is heterodisomy and isodisomy. Heterodisomy is heterozygous, which indicates that meiosis 1 error. Isodisomy is a homozygous, indicates a meiosis 2 error. Okay. And over here, we can consider isodisomy because it's a homozygous, meaning that both of the genes are deleted on the same chromosome. That's why there's a more chance of isodisomy coming up with Prader-Willi. Another one is heterodisomy, is heterozygous, meaning that you have two genes coming from two chromosomes. So if one of them is deleted, then this should not create a very big problem, especially when it comes to the development of Prader-Willi and Angelman syndrome. But if it's an isodisomy, that is in isodisomy, you have homozygous genes. So if one gene or if one chromosome is deleted, that would result in a knockout of both genes in the chromosome because it's a homozygous disomy. That's why in isodisomy there's an increased chance of developing Prader-Willi and Angelman syndrome when compared to heterodisomy. Next question. Does that mean those who previously mutated will be reactive because the offspring will have 0 chromosome inherited in the previous example that you were giving? Yes. Okay. As a result, what happens is if you have a disomy, for example, both genes are coming from the mom, no genes are coming from the dad, and the genes which are coming from the mom is, for example, silenced or mutated, then that would result in 2 chromosomes which are silenced. So it would be incorrect to say that the child has 0 chromosomes. The child will not have 0 chromosomes. The child will have the chromosomes which are silenced or or methylated. Okay, so that's how. Yes, I mean silenced. Yes, so they will be silenced. Next question. If it is silenced, then what is being expressed? Nothing is being expressed. That's the problem. You understand what I mean? Nothing is being expressed. That as a result, these patients have the diseases, have these clinical features. Okay. Next question. If patients who have one parent disomy need methylation to have the disease, yes. If you have disomy, then one of the parents' chromosomes have to be methylated. If there's no methylation, then there's no problem. Any other question? Is everything clear? Again, okay, so can you explain the terms disomy and isodisomy? Disomy basically means 2 chromosomes coming from one single parent. If it's coming from a homologous chromosome, if two genes are coming from a homologous chromosome, it's called isodisomy. If it's a heterozygous disomy, it's called heterodisomy. That is from two chromosomes instead of one single chromosome. This is disomy. Okay, so that's that. For example, once again, this is a disomy. If you have the mom with AA and the father with AA, if there is no disomy, the child will not have an autosomal recessive disease. If it's a disomy, then the child will have, for example, disomy meaning that over here, you have a child who will receive both copies of the chromosomes from the mother, right? So now the child has a very big, a 25% chance of having an autosomal recessive disease. But before, if there was no disomy, then the child will have 0% of developing an autosomal recessive disease. So that's that. That's why we asked you to identify disomy in a patient who has a single parent as a carrier of an autosomal recessive disease. Okay, next one. Only in isodisomy we can have Prader-Willi and Angelman. No, you can have Prader-Willi and Angelman in both isodisomy and heterodisomy, but the chances of having Prader-Willi and Angelman in isodisomy is much, much higher. Okay, any other question? Okay, good. Okay, so thank you so much for your questions. Hope you guys have your confusions cleared out. Okay, what is the chromosome number where there is genetic problems happening in Prader-Willi and Angelman syndrome? Chromosome number 15. Okay. Okay, good. So chromosome number 15, that's that. Now the next thing that we will talk about is modes of inheritance. Okay. When we talk about modes of inheritance, are we all well acquainted with this piece of information? Have we studied this before in our bachelor's? Autosomal dominant, autosomal recessive, X-linked recessive, X-linked dominant, mitochondrial inheritance. Okay, so let's begin with this one. Autosomal dominant. What do we mean by autosomal dominant? When we talk about autosomal dominant disease, can we assume that the genetic abnormality is in the dominant gene, meaning the gene with the capital letter? Yes or no? Yes or no? Yes. Okay. So if I tell you that you have an autosomal dominant disease, is it needed for the child's genes to be homozygous in order to express the disease, or can the disease be expressed in a heterozygous condition? It can be expressed in a heterozygous condition, because even in a heterozygous condition, the gene is the dominant gene will overpower the recessive gene. So can we assume that if one parent is autosomal dominant for a disease, then there is a very high chance, let's say a 50% chance of the child having the disease? Yes? Okay. So how did that happen? Let's break it down. Okay, let's break this down. So we are talking about modes of inheritance, So first of all, the first thing that we are going to talk about is autosomal dominant gene. Before we do that, let me draw a family tree over here. Okay, so circle for mom and box for dad. Okay, are we well acquainted with these two characters? Yes or no? Do we know who they are? Okay, good. Okay. Do we have any student who do not know their names? Can I just get only 1 second, please? Hello? Hello? Okay. So that's that. Okay. John and Jane. Okay, John and Jane. My apologies. I'm really sorry. Give me 1 second. Hello? Yes. Okay, my apologies. I'm really sorry. Okay, so John and Jane. Okay, John and Jane have Jonas, Janie, Jacob. Okay, Jacob over here. Okay. Let's assume there's another one. Okay. All right. Okay, so let's do this. So first of all, our friend Jane has a problem. Okay, this is for Jane. This is for John. Okay, so can we draw up something like this? The 2x2 table. Yes or no? Yes. Okay, good. So if it's an autosomal dominant disease, can we assume that these two are the issues over here? Yes. Okay. So so is there a possibility that out of John and Jane, for example, Janie and Jacob can have the diseases? Yes or no? Yes. Okay. So it will be something like this one. Okay. And if Jacob marries someone else, is there a possibility that if they have two child, they will have the disease in either one of them? Yes. And if Janie marries someone else, is there a possibility that either one of them can have the disease? Yes. Okay. My apologies. Either one of them. Okay. This is a pedigree or a 2x2 table for an autosomal dominant disease. Okay. Why is this important? I'm going to tell you why this is important, okay? Because what would happen is, for example, let's talk about an autosomal dominant disease. For example, the first disease that could come to my mind is VHL, or von Hippel-Lindau syndrome. Von Hippel-Lindau. I don't know why this is the first disease which came to my mind. So you will receive a question in your URL where they will describe von Hippel-Lindau disease, and in your answer options, they will have pedigrees. I'm not sure if you guys have received these questions as of yet, but they will have pedigrees like this, right? Where it will be something like this, right? And like this one. So by looking at the pedigree, you have to understand which one is an autosomal dominant disease and which one is an autosomal recessive disease. Am I clear? Do you understand why it's important to learn the pedigrees? Yes or no? Okay. So what I am going to do is, after we are done with discussing this, I will test you guys by drawing out the pedigrees to see whether you can perfectly identify which one is autosomal dominant, which one is autosomal recessive. Okay. So with that being said, we are done with autosomal dominant. Okay. And autosomal dominant, it's often pleiotropic, that is multiple apparently unrelated effects, and variable expressivity is also there. What is pleiotropy? What is pleiotropy? Plural phenotype. Do you guys remember pleiotropy? What is pleiotropy? What was the disease, the genetic disease that we talked about when we discussed pleiotropy? Phenylketonuria. What was the disease we talked about when we said variable expressivity? Okay, neurofibromatosis. Good, very good. So family history is crucial to diagnosis. Obviously, family history is crucial. And with one affected parent, on average, half of the children will be affected. Do we have any carriers with autosomal dominant disease? Yes or no? Is there any carrier? Carrier? No, there is no carrier. Okay, no carriers. Okay, next one. Next one that we want to talk about is autosomal recessive disease. Autosomal recessive disease. Autosomal recessive disease, they have with two carriers, on an average, 1/4 or 25% of the children will be affected, and half of the children will be carriers, and 1/4 of the children will be neither affected nor the carriers. So instead of, okay, no, you know what? I'm still going to draw this out. Okay, let me draw it out for you guys so that it's easy for you to understand. Okay, so we are talking about the autosomal recessive diseases. Okay, so we're talking about the autosomal recessive disease. Over here, this time, John and Jane, they do not have any diseases expressed. But for some reason, when they have Jacob, Janie, and the two other J kids, okay, what happens is they realize that out of all four of them, suddenly only one child has the problem. So if you see a pedigree or a genetic graph or a tube or a family tree like this, okay, if you see a family tree where two parents are okay, okay, three children are okay, but one child is not okay. One child is not okay. Then can you immediately assume that it's an autosomal recessive condition? Yes or no? Yes. Can you, okay, you can immediately assume it's an autosomal recessive condition. And in the same way, what would happen is, let's say this guy is AA, right? And this guy gets married to someone else who is AA over here. Then is there a possibility that when they have two child, none of them will be affected? Both of them will be okay? Yes? Okay. The only way that this can go horribly wrong is if this woman over here has A and a small letter A. So what would happen is there's AA and AA. Now there is a very big problem. Okay, the very big problem is now the chance of having the problem is 50%. Okay, now the chance of having the problem is 50%. Okay, but this is very rare. This is rare. So the pedigree graph, which I want you to look out for, is something like this. Mom and dad, they're okay. Out of 4 child, one of them is affected. For example, only one of them is affected and the rest of them are okay. This is a pedigree for autosomal recessive disease. Are we clear about this? Okay, good. Okay, so what's happening over here is with two carriers on an average, so we talked about this, most often, autosomal recessives are often due to enzyme deficiencies. So most diseases of autosomal recessive conditions are enzyme deficient conditions. They're usually seen in only one generation. That is, they're usually present in one generation over here. So if I tell you that if I were a patient of cystic fibrosis, is there a possibility that both of my parents are carriers? Yes. Yes or no? Yes. Okay. So that's that. That's that. So commonly more severe than dominant disorders, patients often they present with childhood. So what happens with autosomal recessive conditions are enzyme deficiencies. Enzymes are extremely important in all stages of our life, right? So what happens is that with autosomal dominant conditions, they appear much later in life. But with autosomal recessive conditions, there's a possibility that they can appear much earlier in life. So that's that. There's increased risk in consanguineous families, and the unaffected individuals with affected sibling, unaffected individual with affected sibling has 2/3 the probability of being a carrier. This is the part of the text which got me confused yesterday. Do you guys remember how I made the confusion with the 2/3 of the carrier problem? Yes or no? Yes. So if there's unaffected individual, meaning that if you want to understand the frequency of the carrier with the healthy individual, right? Do you remember how I pointed out that yesterday, if I want to calculate the frequency of the carrier to the healthy individual, then it's 2/3. But overall, the carrier status is 2/4. Okay, we discussed about this yesterday. Does it need any more further discussion? Yes or no? No. Okay, good. Thank you so much. Next one. Next one is X-linked recessive. X-linked recessive. Once again, males or females, who has two X chromosomes? Males or females, who has two X chromosomes? Females, right? I have a friend who scored 250, and to this day, she gets confused whether females have 2X or males have 2X. Okay, I'm not sure if we have any students over here with the same type of confusion, but we have a lot of students who get confused with X's and Y's. And the best way to remember this is, do you guys remember when we studied the reproductive system? Yes, she reads a lot. Okay, that's that. Okay, okay. Do you guys remember when we studied the reproductive system? We talked about the SRY gene on chromosome number Y. Yes or no? Yes. So basically, if we did not have the Y chromosome, then we are all females by default. Do you remember that discussion that every child is born as a female? And why it's very stupid of people to look down on female, because every male was once a female. Okay, so every one of us are born as a female until and unless the Y chromosome comes in and they change the mesonephric and paramesonephric ducts formation. So that's that. Okay, so with that being said, X-linked recessive is this one. The sons of heterozygous mothers have a 50% chance of being affected. Heterozygous mother. What does that mean? That basically means that if this is the mom, right, and if this is the dad, right, the sons of heterozygous, over here, there is no male to male transmission, meaning that if the mother is affected, for example, if you can see over here that the mom over here is not affected and the father for some reason is some sort of a carrier, okay, but in this entire generation there is no one who's affected. I'm going to tell you why. Because over here in this two daughters, they have two X chromosomes. So if one of them is defected, the another one, the healthy one, will overpower the diseased one. As a result, everyone in this family is happy. So in order for this one to, in order for X-linked recessive diseases to happen, then the disease gene is a recessive gene in the X chromosome. So if the mom has a carrier, for example, if the mom is an X-linked recessive mom, then the only person who would be affected is one male child. Because over here, although the mom will transmit this X chromosome to the daughter, but this daughter over here is not affected because she has another X chromosome, right, which can overpower this. But the son over here has only one X chromosome, which is faulty. As a result, the Y chromosome obviously cannot overpower the X. Doesn't matter how healthy it is, because X have their own functions and Y have their own functions. As a result, the son will only be affected. So if we draw a pedigree or a family tree, Okay, so what would we see? What would we see if we draw a pedigree or family tree over here? Then John, Jane, they have 4 child, 1 boy, 1 girl, 1 boy, 1 girl. Over here, only 1 boy is affected. If we keep the diagram like this, then can we confuse this with autosomal recessive disease? Yes or no? Yes? Okay. But what if we were to do this? Because we know that the females are the carriers. Then can we, if we do something like this, now can we assume this is an X-linked recessive disease? Because how, once again, the male is affected. Okay, so what do you look out for in X-linked recessive disease? In X-linked recessive disease, you look out for male affected, females are okay. They're the carriers. Okay, are we clear about this? Yes or no? Okay, once again, let's do something. Okay, let me draw another family tree. Okay. What type of disease is this? What type of disease is this? Okay. What type of disease is this? What type of disease is this? Fast answers. Okay. What type of disease is this? Oh, wait. Did I just marry the 2 siblings? Okay, that is inappropriate. Okay, that is very inappropriate. Okay, this is not Game of Thrones. Okay, my apologies. This is real life where incest is not acceptable. Okay, so my apologies. Okay. Okay, what type of disease is this? Very good. X-linked recessive. Okay, so can we confidently identify all the modes of inheritance? Autosomal dominant, autosomal recessive, till everything we have studied till now? Yes or no? Yes or no? Okay, good. Okay, now if we talk about another disease, can you show again? Yes. Autosomal dominant. Wait, autosomal dominant would have been, let's just look at the ones over here. Autosomal dominant is this one. Autosomal recessive, X-linked recessive. Okay, don't worry, Dr. Karbasi, we will do another test, and over there I will ask you whether you can identify it or not, and only when I'm assured that everyone is perfect in identifying the modes of inheritance pedigree table, then I'll move on. Okay, don't worry about anything. Let's move on to X-linked dominant. Now, what is the problem? The problem is over here, instead of X having the recessive gene, the X has the dominant gene. The X has the dominant gene. If the X has the dominant gene, now is there a possibility for the females to get affected? Because because it doesn't matter right now if the female have 2 X chromosomes, because one of them is faulty, and one of them is faulty and overpowering, that is dominantly faulty. So the another gene cannot overpower that faulty gene. So as a result, now can we assume that both the females can be affected? Yes, if the father is also X-linked dominant. And in the same way, if the mother is also dominant, then you will have 50% affected. So it's transmitted through both parents. The mothers transmit 50% of daughters and sons. The fathers transmit to all daughters, but no sons. So what's happening over here? Okay, so as you can see that over here, if we draw a pedigree, I mean, if we draw a 2x2 table at first, if this is the father who is affected, then since the mother is not affected at all, both the sons are okay, but both the daughters are damaged, or both the daughters are diseased. And in the same way, if the mother has the gene which is affected, then only 50%, that is the son and daughter, will be affected. So if I were to draw something like this, instead of wait one second. Okay, if I were to draw something like this, okay, so this is the mom, this is the dad, okay, and over here, and over here. Okay, if I were to draw something like this, is there a possibility that this could be an autosomal dominant disease? Is there a possibility that, okay, good. So, so there is a possibility that this could be an autosomal dominant disease. Okay. Now, if I were to draw something like this, okay. Right now, let me draw something like this. Okay. So if I draw something like this one over here, and over here, this is X-linked dominant. Now, over here, one child is again affected. Okay. Still, there is a chance that this could still be autosomal dominant. Am I correct? Yes or no? Yes or no? Okay, good. So the only way you can differentiate autosomal dominant and autosomal dominant and X-linked dominance is, for example, the best way UWorld will try to make you understand, because UWorld knows, the people who are responsible for making the questions, they know that autosomal dominant modes of inheritance pedigree and X-linked dominant modes of inheritance of pedigree, they look the same. They look the same. Okay, so what they will do is they will try to make a graph like this. They will try to give you hints that this is X-linked dominant. What they will do is they will create a pedigree like this one. Okay, and they will tell you that the father is affected. Okay, and over here. Okay. Okay. Although this might be an autosomal dominant condition, but can you have a big suspicion that this could also be X-linked dominant? Because as you can see over here, that both the females or both the daughters are affected and the father is affected. Okay, why is there such a big chance for both daughters being affected, but the sons not being affected at all? This should arise a very big suspicion. Okay, so unfortunately, based on this suspicion of yours, you have to come up with the conclusion that if it's an X-linked dominant or an autosomal dominant condition. So what a USMLE will do is they have only one or two questions where they want you to differentiate between autosomal dominant and X-linked dominant. And in most answer options, you do not have autosomal dominant and X-linked dominant together, because they know that USMLE is a very stressful exam and the likelihood of messing up the answers are very high. So what they would do is they would have answer options for autosomal dominant, autosomal recessive, X-linked recessive, and, and mitochondrial inheritance. That's what they would do. So that's the best. That's the best thing I can tell you right now. But even if they have the capability of making questions with autosomal dominant and X-linked dominant together, the thing that you have to look out for is the father being affected and both the daughters being affected. Okay, now there's the chance this is an X-linked dominant disease. Okay, are we clear about this? Yes or no? Okay, good. Let's go back to our text. Oh, I'm sorry. Okay. Okay, so some examples of X-linked dominant diseases are Fragile X syndrome, Alport syndrome, Hypophosphatemic Rickets. Okay, phosphate, which is an X-linked hypophosphatemia. The phosphate wasting is at the proximal tubule, which will have rickets-like presentations. Hypophosphatemic rickets. Okay, I'm not sure if we have talked about this before. If we have not, then this is just a disease where you have loss of phosphates at the proximal tubule, and this is an X-linked dominant disorders. We're not going to study the diseases as of right now because we will use physio for our mnemonics in a second. So that's that. This is X-linked dominant. Next one. Next one is mitochondrial inheritance. Mitochondrial inheritance is extremely easy to understand. Mitochondria, does it come from the father or the mother? Fast answers, please. From the father or the mother? It comes from the mother. If it comes from the mother, can both the sons and the daughters be affected? Yes or no? Yes. But who has the highest chance of passing on the affected chromosome, the daughter or the son? The daughter, exactly. So you will have something like this. I'm not going to draw any more pedigree for this one. The only thing you have to look out for is you will see that the female is always passing on the gene. So you will have something exactly like this one, that you have the mom who is affected, and obviously the mom is giving away the genes. And both the sons and the daughters are affected, as you can see over here, because both of them receive their mitochondria from the mother, right? But when the daughter goes through, has another two child, that only the daughter will pass on the gene and the son will not pass the gene on. This is known as mitochondrial inheritance. The two diseases we have talked about so many times is mitochondrial myopathies and Leber's hereditary optic neuropathy. So let's talk about this. Mitochondrial myopathy, it's a rare disorder. It often presents with myopathy, meaning muscle pain. Over there, you also have lactic acidosis and CNS disorders, which is known as MELAS. MELAS stands for mitochondrial encephalopathy, lactic acidosis, and stroke-like episodes over here. So you have myopathy, lactic acidosis, and you have signs. You have patients with clinical features of syncope. So that's that. The problem is over here, there is secondary to failure in oxidative phosphorylation, and this is extremely high yield. This is the pathology of MELAS. Obviously, if the mitochondria is not working, then you cannot produce ATP. That is, there is failure in oxidative phosphorylation. This is extremely high yield. And another one is you will receive hundreds and thousands of questions in your UWorld or your actual exam where this will be there in your question. That is, if they do a muscle study, then they show that in muscle biopsy, there will be ragged red fibers. Ragged red fibers due to accumulation of diseased mitochondria in the subcirculemma of the muscle fiber. In the subcirculemma of the muscle fiber. So if you ever receive a question about muscle histology, they have ragged red fibers. Whenever you see the word ragged, think about mitochondrial inheritance and mitochondrial myopathy. Okay, are we clear about this? Yes or no? How many times have you guys received questions with ragged red fibers in the muscles and the answer being mitochondrial inheritance? Exactly, right? So that's that. This is this is one of the highest yield associations. This is the most highest yield associations of mitochondrial myopathies, that if they do a muscle biopsy, they will see ragged red fibers. Okay, that's that. Next one. Next one is, this one is not very high yield, although we talked about this in our UWorld notes, that in the Leber's hereditary optic neuropathy is cellular death in optic nerve, which results in subacute bilateral vision loss in teens. Do you guys remember the small piece of notes which we mentioned, the findings in iris that we usually see in Leber's hereditary optic neuropathy? Who remembers that small piece of information? Leber's hereditary optic neuropathy. What did we discuss about Leber's hereditary optic neuropathy in our UWorld notes? Anyone? Anyone? Okay, no one remembers what we discussed about Leber's hereditary optic neuropathy in our UWorld notes. Let me ask, Dr. Jordan remembers. Okay, Dr. Jordan, what do you remember about Leber's hereditary optic neuropathy in our UWorld notes? Okay, craniopapillary something, very close. Okay, so what do you guys have to do about this? Okay, what do you guys have to do about the fact that you guys do not remember the UWorld note? Okay, the best thing that you have to do is read the UWorld note. That is an absolutely right answer. Okay, so this shows that you guys are actively solving questions and you guys are doing very well in your questions, and I'm really proud of you guys. But having said that, there we go. Rapid progressive vision loss with craniopapillary telangiectasia. Okay, Dr. MS. Thank you so much. So can I ask you guys to read the UWorld notes, please? Because we have mentioned a lot of high yield stuff over there, which will actually differentiate you from being a student in the 230s to being a student in the 260s and 270s. Okay, because a lot of high yield informations are in those small piece of notes, because every one of the lines in the notes is a single question. Okay, so can we ask you guys to read the first two pages of the UWorld notes, and can we do a small test tomorrow to see whether you guys have read it or not? Okay. How many pages of UWorld notes do we have? 18 pages, am I correct? 18 pages, right? 20 pages, okay, good. 20 pages. So let's see if we can do 2 pages every day, then we can finish it in 10 days. Then we can finish it in 10 days, okay? Okay, good. How long will it take for you guys to read the UWorld notes? Not even 10 minutes. 10 minutes. Every day, 10 minutes. That's all you need to give, 10 minutes. Okay, so that's that. Okay, so let's talk about the pedigrees. Okay, let me draw a pedigree over here. Okay. Okay. Question before we start? Yes, that's what I was thinking too. Question before we start. Okay. Okay. Oh, you have a question. Okay, so what is your question? Yes, what is your question, Dr. Naud? What is your question? In the autosomal dominant, the shaded circle could be of mom or dad, right? No, the mom is a circle, the dad is the box. Okay. Could you? Yes, if it's shaded, that means they're diseased. Yes. Okay. If they're shaded, that means they're diseased. Okay. Are we clear? Yes or no? Can you please unmute yourself and ask me the questions a bit quickly, please, because we want to get ahead. Please unmute yourself and ask me the question, because my apologies, I cannot figure out what you're trying to ask.
Can you hear me now?
Yeah, I can hear you, Dr. Naud. What is your question?
In case of autosomal dominant, the disease pattern could be either in the mom side or in the dad side, right? That's my
In
question.
autosomal dominant, the disease is on the mom side or on the dad side. Basically, if this is the autosomal dominant one that we're trying to figure out, if it's shaded, then this means that the mom is diseased. If this one is not shaded, this means the dad is not diseased.
Yeah, what my question is, in this case scenario, the shaded case can be also the dad because it's an autosomal dominant disease, right?
How? Because it doesn't. How is for example, this is the mom and this is the dad.
Yeah.
The dad is not shaded. The mom is shaded. That means that this person has they do not have the disease, and this woman, she has the disease.
Okay. Does this graph stand if the other way around is true? The mom not sick, the dad being sick.
Yes.
Okay, that was my question.
Okay. All right. Good. Thank you so much for asking that question and clearing your confusion. Okay. Okay. Now, this pedigree that I have drawn over here, what does this pedigree represent? Fast answers, please. Everyone? Okay, good. Okay. What does this pedigree represent? What does this pedigree represent? Okay. What does this pedigree represent? Okay. What does this one represent? Are you sure? Yes? Okay. So this one is mitochondrial inheritance. This one is not mitochondrial inheritance. Okay, let me see. Did I make a mistake? Did I make a mistake over here? Right. My apologies. Right. You're right. You're right. You're right. You are right. Okay. Okay. No, don't be confused. Every one of them is affected. My apologies. I apologize. Okay. I apologize. I'm sorry. I have so much going on in my head nowadays. I make more mistakes than I used to make before. Okay. I have to remember each and everything. Okay. So, right. Okay. Now it's mitochondrial inheritance. Okay. Okay. If we have a teacher and if we have a student, and if the teacher makes a mistake and if the student corrects the teacher, what does it then what does it say about the student? If we have a teacher and if we have a student, if the teacher makes a mistake and if the student corrects the teacher, what does that mean? We are all learning. No, that means the student is better than the teacher. Okay. This is what that means. Okay, so thank you so much for, you guys have no idea how much I appreciate you guys when you guys correct me. Okay, so that's that. So this is mitochondrial inheritance. Having said that, next time when we X-linked again, okay, X-linked again is X-linked recessive. X-linked recessive. I'm not going to draw the X-linked dominant one, okay, because it will coincide with my autosomal dominant. Okay. That's that. Another one is, and over here, if they have another two child, and this one is affected. This is X-linked recessive. Okay. X-linked recessive. Okay, are we clear about this? Yes or no? Okay, good. Okay. Now, this is the next part of the page which we have to master. Autosomal dominant diseases. Okay. You have to know about all the autosomal dominant diseases and all the autosomal recessive diseases. Okay. And then you have to know about the X-linked recessive diseases. The thing is, you will receive multiple questions in your USMLE step 1, which is a direct question. They will directly ask you, for example, you have a disease such as familial adenomatous polyposis. The question will be something that you have a male who comes to your clinic who has multiple polyps found in colonoscopy, and they have found to be cancerous polyps, and it was present in their previous generations, right? And they will try to make the questions very complicated, but in the last segment of the question, they will ask you a very direct question. That is, what is the mode of inheritance of this disorder? So first and foremost, you have to identify that they are trying to ask you about familial adenomatous polyposis, and then you have to identify directly whether it's autosomal dominant and autosomal recessive. So is this a matter of memorizing or not? Yes or no? All these diseases, yes or no? Okay. If yes, it is a matter of memorizing. it is a matter of memorizing. I'm going to tell you why, because you have to memorize all these things which are represented as being as autosomal dominant diseases. Okay. So what if I were to tell you that we have this friendly resource over here, which I've been waiting for so long to use? It is hard. I know it's hard, but thank God for physio, because we can use this too. It will only take us 4 minutes. Okay, so this is what we will do. First and foremost, when we use physio, it's a 4-minute video which we will watch. After that, we will use the picture mnemonic, and one of you guys are responsible for taking a picture of the picture mnemonic and posting it in the group. Okay, so who would that person be? Who would that kind person be? Who can take the responsibility of taking the picture? Anyone? Because I cannot take the picture, because if I take the picture, then I would be then my account will be deleted. You understand what I mean? I can. Okay. So make sure that you take a picture, which is a screenshot. Okay. Try to take a screenshot, and I will ask you, and I will tell you exactly when you can take the screenshot. So you do that. Okay. And Dr. Jordan, thank you so much. And please be kind enough to upload that in the discussion group. Okay. So that's that. Okay. So without further ado, let's begin. Okay. Very excited right now because I've been wanting to share this with you guys for so long. Okay.
Welcome to section 6.1 of genetics. In this section, we'll be memorizing the autosomal dominant disorder.
1 second.
Let's get started. First off, here is the
One second, please. Okay. Okay.
Welcome to section 6.1 of genetics. In this section, we'll be memorizing the autosomal dominant disorders. Let's get started. First off, here is the beautiful outdoor fall background where this scene will take place. Next, we'll show an old broken down car with dominoes hanging from the rear view mirror. I know, you usually see dice hanging from the mirror, but dominoes look kind of like dice and it fits the story better. The dominoes will represent autosomal dominant diseases. So dominoes for autosomal dominant. The old broken down car will help you remember hypertrophic cardiomyopathy. So car for cardiomyopathy. The car belongs to a hunter, so we'll show a hunter next to him. The hunter is currently hunting with his rifle, so this should help you remember Huntington disease. Notice that he's wearing patched up clothing. He also has an old broken down car. Both of these ideas allude to the idea that he's poor. Just think of this old hunter guy who's unable to pay for his bills, so he drives out to this forested area in search of food. So poor for acute intermittent porphyria. Next, notice that he's hunting this hippo. He's pretty desperate for food because it's obviously pretty shady for this guy to be hunting hippos, but he doesn't seem to care too much. He's determined to get himself some hippo meat. The hippo should help you remember von Hippel-Lindau disease. Also notice that the hippo has very prominent and swollen lips. The word lip is in polyposis, so this refers to familial adenomatous polyposis. His lips are swollen because he's been eating these red berries that look kind of like red blood cells. Unfortunately for our hippo friend, he's allergic to the berries, hence the swollen lips. These small, round, spherical berries represent hereditary spherocytosis. The tree has acorns on it. It's fall and the acorns have become ripe, so they've started falling off the trees. So notice that one of the acorns has fallen off the tree and bounced off the hunter's head right here. The acorns will represent achondroplasia. Next, we see some leaves falling to the ground in this fall weather. The leaves signify Lee-Fraumeni syndrome, so leaf for Lee-Fraumeni. Notice that the river is now filled with a bunch of little spots. These represent oil or fat, and the river represents the vascular system. Together, these should help you think of familial hypercholesterolemia. Okay, notice that the river is overflowing onto the ground. Von Willebrand factor normally binds to the collagen during endothelial damage and prevents hemorrhage. Therefore, a leaky river should make you think of a leaky blood vessel, which is basically what happens in patients with von Willebrand. these two guys tubing down the river. They were pretty stoked for a day out of the office and decided to go tubing down this awesome river. Up to this point, they had been enjoying the thrilling adventure, admiring the fall leaves and all the acorns on the ground. However, right as they come across the bend, they suddenly become scared about this hunter guy, who seems to be a shady character. The tube represents tuberous sclerosis. Also notice that this guy has a pretty awesome looking spiderweb tattoo. This is likely the last time he can show it off to his friends because it will soon be too cold. This should make you think of hereditary hemorrhagic telangiectasia, because in this disorder, patients develop telangiectasias, which look kind of like spider webs. Also notice that their arms are very long and lanky, which is a characteristic finding seen in Marfan syndrome. This is a good thing for them because they can maneuver around and paddle extra fast with those long arms. Hopefully they'll be able to avoid our crazy hunter character and avoid the gunfire. So the long arms represent Marfan syndrome. Notice on the very ends of their paddles are little frayed fibrous edges right here. They're also going through somewhat of a financial crisis like our hunter friends, so they of course had to bring old used paddles. The fibrous ends of the paddles represent neurofibromatosis. Both neurofibromatosis type 1 and type 2 are autosomal dominant conditions. Next we'll add a third guy who has unfortunately fallen off the tube in this perilous adventure. They all seem pretty scared of this hunter guy, so this third man jumped off to avoid the gunfire, but the old man isn't a very good aim and accidentally shot him in the leg. Maybe it was the fact that the acorn bounced off his head and messed up his aim, or that he's just a terrible aim. Either way, our hunter friend is now in some serious trouble with the law. Luckily for the injured man, he only has a leg wound, so he should survive. So the three men represent Men syndrome. Notice that the man who took the bullet has a fragment of bone sticking out of his leg. This should make you think of Osteogenesis Imperfecta. Recall that Osteogenesis Imperfecta is a genetic bone disorder that results in decreased production of type 1 collagen, resulting in fragile bones. So the fractured bone here from the bullet should hopefully help you connect these ideas. Finally, notice that he also has a little bit of muscle hanging off of his leg. The muscle should make you think of myotonic muscular dystrophy. On a brighter note, the hippo seems pretty happy that he narrowly escaped death.
Okay, so thank you for watching this small piece of cartoon over here. And this is your picture mnemonic for the autosomal dominant disorders, okay? The dies for autosomal dominant, hypertrophic cardiomyopathy, achondroplasia, Huntington's disease, familial hypercholesterolemia, Marfan syndrome, neurofibromatosis, osteogenesis imperfecta, von Hippel-Lindau, myotonic muscular dystrophy, familial adenomatous polyposis, right? Did we understand the disorders, the names of the disorders from this picture? Yes or no? Guys? Yes or no? Okay, okay, most of them, but okay. So without even reading the first aid, without even reading, can you add me in the discussion group? Of course, I can add you in the discussion group, no problem. And so isn't this picture mnemonic helpful if you want to remember all the disorders? Because if you close your eyes, the only thing you can see is the picture instead of looking at the text in first aid, right? But when you go and look at the text in the first aid, then you can see that they have covered almost everything over here. Okay, so, okay, Dr. Jordan has just posted it. Thank you. So let's see if we have covered everything. Okay, let's see if we have covered everything over here. So what are the autosomal dominant disorders? Achondroplasia, autosomal dominant polycystic kidney disease, familial adenomatous polyposis, hypercholesterolemia, so far and so forth, Hereditary Spherocytosis, Lee-Fraumeni syndrome, Marfan syndrome, men, myotonic muscular dystrophy, neurofibromatosis, right? These are the autosomal dominant diseases. Okay. So would anyone be kind enough to take the chance of mentioning some of the high yield autosomal dominant diseases in the chat box? In the chat box. Anyone in one go? How many autosomal dominant disorders can you mention in one go? Guys, anyone? Would anyone be kind enough to mention the autosomal dominant disorders by remembering the picture of physio? Anyone? Okay. 8 to 10 max. Okay, good. We can try. Okay, so how about this? Let's watch the video for the autosomal recessive, and then we can see if we can differentiate or not, okay? Okay, how about that? Okay, good. The picture that is posted in the group, okay, what you guys can do is you guys can save that picture, and when you guys do a revision of first aid, that will help you to read these disorders very easily. Okay, was the picture mnemonic helpful or not helpful? Did you guys like it or not like it? Because if not, then we cannot use physio. We can just discuss this by ourselves. I personally feel it's really helpful. I'm going to tell you why. Because if you close your eyes and try to think about the diseases, the picture will come. Please mute yourself while watching the video. Of course I will. Okay, my apologies. I will mute myself too when we are watching the videos. Okay, so next one is, let's begin with autosomal recessive disease. Okay, let's begin with autosomal recessive disease. What are the autosomal recessive diseases? Okay.
Welcome to section 8 of the disorder. Fibrosis or CF creates chloride in. You can see chloride retention. Welcome to
Okay, 1 second. Okay, so I think I understand what they did. Okay, so let me tell you what they did. So what they did was, okay, so all the diseases which you have just read, they were autosomal dominant diseases. Am I correct? Yes or no? Yes or no, right? Okay. So what if I were to tell you that all the other diseases which are not autosomal dominant, they're all autosomal recessives? That's exactly what they did. That's why they do not have any other picture on autosomal recessive diseases. You understand what I mean? But you can also coincide with X-linked disorders. So what they did was they have one for autosomal dominant and they have one for X-linked recessive disease. So let's watch the X-linked recessive diseases in order to differentiate properly. Yes or no? Okay. Can we watch the X-linked recessive diseases? Because we have to learn this anyways. So instead of watching the autosomal recessive diseases, let's watch the X-linked recessive diseases. Okay. Okay, perfect.
Welcome to section 7.1 of genetics. In this section, we'll be memorizing X-linked recessive diseases. This scene will take place in an old couple's home during the Christmas season.
My apologies, Dr. Iman, if I unmute myself, or if I mute myself, then you guys will lose the voice. I'll try to make sure everything is as quiet as possible. Okay. Okay. I would make sure everything is as quiet as possible. Okay.
Section 7.1 of genetics. In this section, we'll be memorizing X-linked recessive diseases. This scene will take place in an old couple's home during the Christmas season. First, we'll show a nice decoration on top of the Christmas tree. In this story, there will be an X on top of the tree, kind of like a star. The X should help you remember that this scene is about X-linked recessive diseases. Next, we'll add ornaments to the tree to make the scene a bit more festive. This should help you remember ornithine transcarbamylase deficiency. So ornaments for ornithine. Okay, I should mention that this is taking place in a retired hunter's home, hence the antlers that are hanging on the wall. This trophy should make you think of X-linked adrenoleukodystrophy. It's next to the X on the tree, and adrenoleukodystrophy has the word trophy in it. So X and trophy should make you think of X-linked adrenoleukodystrophy. The retired hunter also has a bunch of hunting trophies on the fireplace mantle that are from his early hunting career. These trophies should make you think of Duchenne and Becker muscular dystrophy. So again, trophy for dystrophy. Okay, notice that this retired hunter has a bunch of guns on the wall and that he's reading a magazine. The trophies, gun case, and him sitting in the chair should all make you think of a retired hunter. This is different from the picture of autosomal dominant conditions where we showed the hunter actively hunting. So in this case, the retired hunter will represent Hunter syndrome. Huntington disease and Hunter syndrome can be a difficult point to memorize, so hopefully you'll be able to keep this straight by picturing the retired hunter. Notice that he's now shown with reading glasses. Most older people have a harder time reading without their reading glasses, so this seems to fit the story nicely. The glasses are clear, just like patients with albinism lack pigment. So the clear glasses should help you recall ocular albinism. He's also reading a magazine about hunting called Brute Force. The name of the magazine ties in nicely with the hunter theme. Just imagine that it has pictures and articles about hunting, guns, and the military. Brute Force is a title that sounds like it's related to the military, so this should make you think of Bruton agamma-globulinemia. Okay, now we'll add a bottle of whiskey next to his chair that he's been enjoying while reading his magazine. Just imagine that it's the end of a long day and he's finally sitting down to read his hunting magazine and enjoy. He will represent Wiscon-Aldrich syndrome. Like most hunters, this hunter has a dog that has helped him become the successful hunter he is today. He likes his loyal companion to stay close, so we'll show a dog on a leash who is tied up to the chair. The leash should help you remember Lesch-Nyhan syndrome. Like most hunting dogs, this one has a lot of energy and needs to be tied up on a leash because he's super hyper and would otherwise run around the entire house making a huge mess. So hyper dog for hyper IgM syndrome. We'll also show the dog eating some Philadelphia cream cheese with an apple and a banana. Our old hunter guy has had a long day and he's too interested in his magazine to pay attention to what his dog is eating. So just imagine that the dog got into the garbage and is now snarfing down the food, hoping he can finish it off before the hunter character will notice. The Philadelphia cream cheese sounds kind of like hemophilia, so Philadelphia represents hemophilia. The apple and banana should help you remember A and B. Outside of the house, we'll show some skid marks on the road. I like to imagine that the hunter came flying home in his car after a long day, so the skid marks are from his eagerness to get to his magazine and dog. The skid marks will represent severe combined immunodeficiency, or SCID. A monk may not fit perfectly with the holiday season, but we like to think of him attempting to share his religious beliefs with this hunter. So the monk represents Menkes disease. There will also be a fire hydrant next to the monk. The hydrant sounds like dehydrogenase, so this will represent glucose 6-phosphate dehydrogenase deficiency. Finally, we'll add the hunter's wife, who is crocheting some fabric and enjoying the warmth next to the fireplace. So the fabric represents Fabry disease.
Okay, so did you guys understand the video? Yes or no? Okay, Dr. Jordan, did you succeed in taking except Wiscon-Aldrich? Wiscon-Aldrich. Dr. Jordan, did you? Yes. All right. Thank you so much. Okay. Wiscon-Aldrich is for the whiskey bottle over here. Okay. It's for the whiskey bottle over here.
On agamma-globulinemia. Okay, now we'll add a bottle of whiskey next to his chair that he's been enjoying while reading his magazine. Just imagine that it's the end of a long day and he's fine.
So at the end of the picture, when you have this whole mnemonic, then this represents every X-linked recessive disorders. Okay. Now, my question to you guys is, if you guys know about the autosomal dominant diseases and the X-linked recessive diseases, and then can we assume that all the other diseases are autosomal recessive mostly? Yes? Okay. Okay. So that's what it is. So let's look at the autosomal recessive diseases. The autosomal recessive diseases are oculocutaneous albinism, autosomal recessive, polycystic kidney disease, cystic fibrosis, Friedreich's ataxia, glycogen storage diseases, hemochromatosis, cardiogenic syndrome, then mucopolysaccharidosis, phenylketonuria, sickle cell anemia, sphingolipidosis, thalassemias, and Wilson's disease. Basically, these are all the diseases which are not present in the autosomal dominant ones. Okay, X-linked dominant. For X-linked dominant, the two diseases that you have to think about is Fragile X syndrome and hypophosphatemic rickets. Fragile X syndrome, hypophosphatemic rickets, and Alport syndrome. These are only the three things that you have to remember about X-linked dominant. Fragile X syndrome, hypophosphatemic rickets, and Alport syndrome. Okay. So now let me ask you guys very fast questions. First of all, we have Ornithine transcarbamylase deficiency. What type of inheritance is this? Fast answers, please. Okay, how about cystic fibrosis? Friedreich's ataxia. Okay, von Hippel-Lindau disease. Okay, Marfan syndrome. Familial hypercholesterolemia. Tuberous sclerosis. Huntington's disease. Multiple endocrine neoplasia. Wilson's disease. Very good. Wiscon-Aldrich syndrome. Menkes disease. Menkes disease. Lesch-Nyhan syndrome. Fabry disease. Duchenne's muscular and Baker's muscular dystrophy. Alport syndrome. Alport syndrome. Hypophosphatemic rickets. Hypophosphatemic rickets. Okay. G6PD deficiency. How about hemophilia A and B? Sickle cell anemia. Okay. Okay, that is absolutely wonderful. So how do you guys feel? Do you guys feel confident that if you come across a disease, you can identify the diseases perfectly? Okay. So now can we assume that it's not actually as difficult? It's just a method of learning how you learn something. Okay. How would this be different if we asked you to blindly memorize the diseases? It would be difficult, am I correct? Yes. If I asked you to blindly memorize the diseases, but if you have, I keep on talking about the memory championships, okay, because I was watching a lot of memory championships when I was preparing for my step one, and a lot of memory champions, what they do is the brain is wired to remember pictures and videos. So that's why we remember movies way more than we remember books. Okay. If you, for example, like if you have a book and if you have a movie, although the movie will never do justice to the book, but it's easier to remember the scene from a movie, right? So that's the same thing which you can do too. And that's why we use the physio. Hopefully that was really helpful. So the next time you want to do a revision of all the disorders, what you have to do is look at the pictures that has been posted in the discussion group. And when you see those two pictures in the discussion group, hopefully, which are posted by Dr. Jordan, thank you again. Just try to remember if you can identify all the diseases from over there, and all the other diseases which are not there are either X-linked dominant or autosomal recessive. And then once again, X-linked dominant are three, which is Fragile X, Alport syndrome, and hypophosphatemic rickets. Okay, at least for the ones which are high yield for your USMLE step one. Okay, so with that being said, can we take a small break before we begin cystic fibrosis? Or do you want to finish cystic fibrosis right now? Because I want to talk about cystic fibrosis and I want to take my time with this because this disease is extremely high yield. Okay. Okay. And then we have dystrophies. Okay. Okay. All right. No problem. This is what we're going to do. Okay, so let's take a break for 15 minutes and let's come back. Let's come back at 11:05. Okay, is everyone back from their break? Is everyone back from their break? Okay, good. So we are done with understanding the modes of inheritance, the differentiating between the autosomal dominant, recessive, and X-linked recessive and X-linked dominant disorders, imprinting and disomies. And now we are going to jump into a very high yield disorder, that is cystic fibrosis. Okay, so what we are going to do is we are going to study this step by step, and that's what we're going to do. Before we jump into the text of first aid, what I want to do is I want to talk about cystic fibrosis on a separate blank page first, and then I want to go to the text in first aid and see if we have covered everything. Okay, so that's that. Okay, so what we are going to do is we are going to talk about cystic fibrosis. First of all, if we talk about cystic fibrosis, what type of a disease is cystic fibrosis? Is it autosomal dominant, recessive, X-linked recessive, X-linked dominant? What type of disease is this? Okay, it's an autosomal recessive disease, and the defect is in a gene. Okay, the name of the gene, I will tell you the name of the gene in 1 second. Before that, how many letters are in this word? C-Y-S-T-I-C, cystic F. How many letters are here? 7. So where is the genetic defect, on which chromosome? What is the chromosome number? The chromosome number is numbers 7. Okay, the chromosome number is 7. Okay. Now, on this chromosome number, that is the 7th chromosome, there is a gene. The gene which is defected is CFTR. CFTR gene. So if there is a genetic defect, can we assume that there is a defect with the protein synthesis? Yes or no? Yes. So the defect with the protein synthesis is, what happens is, for this autosomal recessive defect in CFTR gene on chromosome number 7, you have a defect which results in the deletion of phenylalanine at position number 508. You have a deletion of phenylalanine at 508. This is not extremely high yield, but it's sort of high yield. But then again, there's nothing about cystic fibrosis which I want to leave untouched, okay, because cystic fibrosis is a very high yield disease. So this is the genetic pathology. Are we all clear about this? If the next time, if anyone asks you, what is the mode of inheritance, what is the gene, what is the chromosome, and which amino acid is affected, can you answer them with absolute confidence? Okay. Okay, good. Next one. Next one is, what is the pathology? Next one is, what is the pathology? What is the pathology of cystic fibrosis? The pathology of cystic fibrosis is that the gene, the CFTR, it is responsible for giving rise to a protein, which is an ATP gated. Remember that it's an ATP gated. This is a very high yield question in NBME, that the cystic fibrosis gene is responsible for the chloride channel. Is the chloride channel ATP gated, ligand gated, or any other answer option? The answer is ATP gated. It's an ATP gated chloride ion channel defect. Chloride ion channel defect. So what happens is this chloride ion channel, what this does is, if you look at this picture over here, this is the normal one. So what happens is the chloride ion channel, it secretes the chloride in the lungs and the GI tract. the chloride ion channel, that it secretes the chloride, and it secretes the chloride in the lungs and the GI tract, and it reabsorbs the chloride in the sweat duct. So what's happening over here? First of all, it's responsible for secretion of chloride. Another one is in the sweat duct, it is responsible for reabsorption. It is responsible for reabsorption. So if I tell you that patients with cystic fibrosis, they have a high chloride content in their sweat, is that a possibility or not? Because since the chloride ion channel in the sweat duct is responsible for reabsorption, so since the chloride is not being reabsorbed, so can we conclude that the sweat in cystic fibrosis patient, they are high in chloride? Okay. So what happens is the mutation, it results in the formation of a misfolded protein. The protein is misfolded. As a result, if there's a misfolded protein, do you remember how we told you that the rough endoplasmic reticulum was responsible for secreting the secretory proteins? Yes or no? Yes. So if a protein is misfolded, do you think the rough endoplasmic reticulum will release a protein which is misfolded into from out of the cell? Yes or no? Will they secrete the misfolded protein, or will they retain the protein? They will retain the protein. Rough endoplasmic reticulum, they are only responsible for secreting the proteins which are proper and which can have their proper functions. So what happens is, if the protein is misfolded, the rough endoplasmic reticulum will not secrete the protein. So what happens is, this protein, it remains or retained in rough endoplasmic reticulum. It is retained in rough endoplasmic reticulum. As a result, when it is retained in the rough endoplasmic reticulum, we can immediately assume that it's not released. If it's not released, then what happens is there is decreased chloride and water secretion. Okay, there's decreased chloride and water secretion, and as a result, okay, let me just write this down. There is decreased chloride and water secretion, right? And as a result, what happens is you have the as a result, what happens is there is increased chlorides inside the cell. If the chloride is not secreted, can we assume the chloride is retained inside the cell? Yes or no? Yes. And is chloride a negative ion or a positive ion? It's a negative ion. So if the chloride is retained inside the cell, can we also say that the sodium is also retained, or there's intracellular shift of the sodium? Yes or no? Because then the negative ion will attract the positive ion. Okay. So what happens is, as a result, the intracellular salt content is high. If I tell you right now that the intracellular salt content is high, for example, this is the cell and this is the secretory area, right? This is the secretory area, and this is the cell. So what happens is, if there's high amount of salt in the cell, can the water shift from the secretory duct inside the cell? Yes or no? Yes. No? Why? Why not? Yes. Okay. Yeah. So what's happening, if there's shifting of water inside the cell, now can we assume that the secretory duct, they have a very thick and viscous mucus? Yes. What happens when there is thick viscous mucus? There is decreased clearance of that mucus. So this will result in predisposition to what? Fast answers, please. This will result in predisposition to what? Thick mucus. Fast answers. Predisposition to? That's not the word I'm looking for. Predisposition to infections. There we go. This will result in predisposing to infections. Okay. Stasis, then infection. Okay, so infection, that's that. This is the pathology of cystic fibrosis. Are we clear about this? step by step, everything. This is the J, this is the gene, this is the pathology. Okay, now how do you diagnose a patient? Well, whenever we learn about a disease, we have to learn about the pathology, clinical findings, right? And diagnosis, and then complications and treatments. Okay, so let's talk about the diagnosis. Okay, I will repeat the steps once again when we read from the text. Okay, just bear with me over here. Let's finish this. Diagnosis. Let's talk about diagnosis. How do you diagnose cystic fibrosis? First of all, the ATP gated chloride ion channel, they are responsible for secreting chloride and reabsorbing chloride from the sweat glands. Am I correct? Yes or no? These are the two functions. So if there is decreased chloride functions in the sweat gland, can we assume that the sweat has a high chloride content? I'm asking the same question again because I want to incorporate this knowledge in your head. Yes. So number one is, if we use a drug, for example, pilocarpine. What type of a drug is pilocarpine? Pilocarpine. What type of a drug is pilocarpine? Okay. Will this result in vasodilatation or vasoconstriction? Okay. If there's dilatation, can we assume that pilocarpine will cause increased sweating? Increased sweating? Yes. So if in pilocarpine, if we give pilocarpine in these patients, which is an antimuscarinic, and this results in increased sweating, then can we assume that if we test the sweat of these patients, the chloride concentrations of cystic fibrosis patients will be higher than normal? Yes, and the answer is yes, so they will be higher than normal. And another thing is, this can also cause contraction alkalosis. Do you guys remember contraction alkalosis? What happens in contraction alkalosis? Do you guys remember, if we lose excess water, then renin, angiotensin, aldosterone is activated? And what happens when that's activated? It works on, where does it work on? The renin, angiotensin, to cause contraction alkalosis? On which part of the renal tubules does it work? It works on the proximal convoluted tubule to increase bicarbonate reabsorption, right? So contraction alkalosis can happen, and at the same time, there could also be hypokalemia, but that's not the one which we are very concerned about. We are concerned about increased chloride sweat content. Okay, and another one is, if we do chest X-ray of a patient with cystic fibrosis, chest X-ray, can we find signs, symptoms of bronchiectasis? Yes. Okay. Then then another one, since we are talking about diagnosis, then another one in CBC, high chance of infection, so increased WBC, right? Even the temperature might be high if the patient has infection. Then what else? What else can we find? If we do an electrolyte examination, over there we can find hypokalemia, again due to renin, angiotensin, aldosterone. Then what else? What else? What else can we find? If we study the sperm count, can we see decreased sperm count? I mean, decreased sperm motility. Decrease sperm motility. Okay, and decreased fallopian tube motion. Okay. Okay. Absence of vas deferens. Where do we see absence of vas deferens? Cystic fibrosis. So, absence of vas deferens. Decreased sperm motility is also seen in cystic fibrosis, but this is more common in primary ciliary dyskinesia. in absence of vas deferens is more common in cystic fibrosis. But to be honest, sperm motility is also decreased. I'm going to tell you why, because there's thickening of the sperm content, right? So that's that. Okay. Next, can we also study? Can we also study the pancreatic enzymes? Will there be normal pancreatic enzymes, or will there be pancreatic insufficiency? Pancreatic insufficiency. Pancreatic insufficiency. If there is pancreatic insufficiency, can we assume that there is decreased absorption of fat-soluble vitamins because the lipid is not broken down properly, right? So decreased fat-soluble vitamins. Okay. Another one is in a newborn. Can we assume that if the newborn has meconium ileus, this is a high suspicion for cystic fibrosis? Okay. Okay. Then what else are we missing out? What else are we missing out? Sodium level. Sodium level, to be honest, it doesn't fall as much to the point that the patient has hyponatremia. Okay. But it does fall, but the electrolytic imbalance is basically hypokalemic electrolytic imbalance. Okay. Another one is patients, they also have a tendency of having nasal polyp. If you have a young child with nasal polyp, did you study cystic fibrosis from Pathoma, where Dr. Sattar mentions that if there's a young child with nasal polyp, there's a high possibility for development of cystic fibrosis. Okay, nasal polyp over there. Okay, now, what do we do if we want to treat the patients? What do we do if we want to treat the patients? Okay. How do you treat a patient? First of all, let's talk about the lung secretions. Let's talk about the lung secretions. Okay. Okay. So if I tell you that you have to treat the patients with antibiotics, and we all know that it's a very okay, treatment. First and foremost, what do we do to treat the patients? First of all, if the patients have a chest infection and bronchiectasis, we can give them antibiotics. That's that. But if we want to target the mucus that is being produced over here, then what we can do is, can we give N-acetylcysteine? Yes or no? N-acetylcysteine, which will break down the mucus. It's a mucolytic. Okay, so N-acetylcysteine. Do you guys know any other places where we can prescribe N-acetylcysteine? Where do we give N-acetylcysteine? Where do we give N-acetylcysteine? Antidote for paracetamol toxicity. Okay. Then, have you guys ever seen a patient undergo chest physiotherapy? Chest physiotherapy. Okay. Chest physiotherapy, which will help get rid of the excess mucus. It's a type of physiotherapy where you can watch a video on YouTube on how they do it, but it's not very high yield, but chest physiotherapy is also done to get rid of the mucus. Then we can give albuterols, right? But I don't want to talk about albuterols, okay? We know that we can give antibiotics, albuterols, right? That's that. Next thing that I want to talk about is hypertonic saline. Hypertonic saline. Why do we give hypertonic saline? Because we know that the cells, they have a high amount of salt inside them. So if we can provide if we can provide a saline which is hypertonic, then the excess water that is inside the cell will move from the cell inside the secretory duct. So if we can give hypertonic saline, not via IV, but we can give hypertonic saline as a humidifier, this will facilitate mucosal clearance. This will facilitate mucosal clearance. And along with this, I want to talk about another high yield drug, that is ivacaftor. Okay, I want to talk about ivacaftor. What is ivacaftor? The ivacaftor, this is a type of a drug which is combined with another drug, that is lumacaftor. Lumacaftor and ivacaftor. These two drugs, they are responsible for correcting the misfolded protein. The misfolded protein that is retained in the rough endoplasmic reticulum is corrected with lumacaftor and ivacaftor. Okay, lumacaftor and ivacaftor. Okay, so that's that. And then again, we can also give all the other drugs. Are we clear about our discussion on cystic fibrosis? Are we clear about our discussion on cystic fibrosis? Okay. Let's talk about the genetics very quickly. What is the name of the gene that is affected in cystic fibrosis? CFTR. What is the chromosome number? What is the chromosome number? Chromosome number 7. Very good. What is the amino acid deletion? Phenylalanine. Good. Next one is the pathology. As a result of this type of problems, what is the primary issue with the, okay, CFTR? Which type of protein does it code for normally in a normal patient? Fast answers, please. Which type of protein do they code for normally, CFTR? Guys, which type of protein does CFTR code for? ATP, gated, chloride, ion channel. Very good. So what happens if there is a deletion of phenylalanine? What is the pathology? What is the pathology? Misfolded protein, which is retained in the rough endoplasmic reticulum. As a result, we have increased chloride in the sweat and decreased chloride and ion secretion. If there is decreased chloride ion secretion, then you have increased sodium, which is also retained, and increased salt inside the cell results in water retention. And when water is retained, this results in thick mucus. This thickening of the mucus is known as the type of mucus which we see in cystic fibrosis. Okay, diagnosis. What is the number one diagnostic test that we can do to diagnose cystic fibrosis? Sweat by pilocarpine. Very good. Then we can also do chest x-rays, pancreatic enzyme study, vas deferens to see if it's absent or not absent, thick cervical mucus to see that, sperm motility, sperm count, azoospermia is also there, nasal polyp is there, right? All those things. Treatment-wise, what is the name of the combination drug which is responsible for treating the misfolded protein or correcting the misfolded protein? Fast answers, please. Cafters, lumacafters and ivacafters. Okay, lumacafters and ivacafters. Okay, then what else? Another thing is, if we have a patient with meconium ileus in newborns, then what is the diagnosis? Cystic fibrosis. Okay. Now let's go to our text. Okay. Let's go to our text right now. Let's see if we have studied everything. Okay. 1 second. My apologies. It's just taking a bit of time. Okay. Okay, so this is the text for cystic fibrosis. First of all, the genetics. This is an autosomal recessive disease, the defect in CFTR gene on chromosome number 7, high yield, commonly a deletion of phenylalanine 508, high yield, most common lethal genetic disease in Caucasian people. Okay, the pathophysiology is that they gate for an ATP-gated chloride ion channel. This is high yield. That secretes chloride in the lungs and GI tract and reabsorbs chloride in the sweat gland. Most common mutation results in misfolded protein. Protein is retained in rough endoplasmic reticulum and not transported to cell membrane, causing decreased chloride and water secretion. This results in increased intracellular chloride, results in compensatory increase in sodium reabsorption via epithelial sodium channels, ENaC. This results in increased water reabsorption, abnormal thick mucus secreted into lungs and GI tract. This increases the sodium reabsorption, also causes negative transepithelial potential differences. As a result, that's why the salt keeps on moving from the secretory duct to the cells. Diagnosis. Diagnosis is if we try to do a pilocarpine-induced sweat test, we will see there's increased chloride ion. This can also present with contraction alkalosis and hypokalemia due to what we have just discussed before. That is, you have angiotensin, right? Which goes and works in the proximal convoluted tubule to increase bicarbonate reabsorption and retention. Because of ECF, water and sodium losses via sweating and concomitant renal, potassium, and hydrogen wasting. It increases the immunoreactive trypsinogen, which is the newborn screening. This is something which we did not discuss. Increase in immunoreactive trypsinogen. Increase in immunoreactive trypsinogen. What happens is that in newborns, the trypsinogen is increased. Okay, what happens is when there is decreased secretion from the pancreatic enzyme, when there's decreased pancreatic secretion, this results in increased formation of trypsinogen, which stays as trypsinogen instead of being activated to trypsin, because trypsin is activated when all the other enzymes in the pancreas are secreted together. So this increase in trypsinogens, they are pretty diagnostic, and they are used as a screening test to see if there is pancreatic insufficiency as a newborn or not. If there's pancreatic insufficiency even as a newborn, then it's highly suspicious of cystic fibrosis. So that's how they use increased trypsinogen to screen for cystic fibrosis. But then again, the most ideal test that we perform is a pilocarpine-induced sweat test. So that's that. Complications are recurrent infections of recurrent pulmonary infections by staph, Pseudomonas infections, allergic bronchopulmonary aspergillosis. So these are the three common microorganisms, that is Staph aureus, Pseudomonas, and Aspergillus. Chronic bronchitis and bronchiectasis, which results in the formation of reticulonodular pattern on X-ray and opacification of the sinuses. Do you realize that our sinuses, they are filled with mucus, right? So if there's thickening of the mucus, can we assume that there's a high possibility for development of sinusitis? Yes or no? Okay, so that's that. Next one is pancreatic secretions are also decreased. So there's pancreatic insufficiency. As a result, you have decreased decreased lipases. So if you have decreased lipases, this results in malabsorption with steatorrhea, decreased fat-soluble vitamins A, D, E, and K. Then this can also cause biliary cirrhosis and liver diseases. And meconium is present in newborns, that is decreased excretion of meconium. That's that. Infertility in men. There's absence of vas deferens. Spermatogenesis may be unaffected, but sperm motility could be affected because there's thickening of the mucus, so that's that. But sperm motility is more decreased in primary ciliary dyskinesia. And in cystic fibrosis, you have absence of vas deferens. Don't mix those two up. And in women, what happens is there's amenorrhea. Along with this, the cervical mucus is also thickened. And this thickening of the cervical mucus prevents fertilization and implantation. So that's why females also have infertility. And if you have a young child with nasal polyps and clubbing, and with nasal polyps especially, then this is a pretty suspicious of cystic fibrosis. So nasal polyp is there. And there's also clubbing of nails because the patients have bronchiectasis. That's that. Treatment is multifactorial treatment. You give chest physiotherapy, albuterol, aerosolized Dornase alpha DNase, which is responsible for breaking down the mucus along with hypertonic saline. Azithromycin is used as an inflammatory agent. We use Zmax, right, as the primary drug to treat lung infections. So azithromycin is there. Ibuprofen slows the disease progression, although no one knows why. Ibuprofen works like how it works, and no other NSAID works in cystic fibrosis. So this is sort of an anomaly, although it's not very high yield, so you don't have to worry about it. The highest yield one is combination of lumacaftor and ivacaftor. Lumacaftor and ivacaftor. Lumacaftor will correct the misfolded protein, and ivacaftor will open the chloride channel and improve the chloride transport. Okay, so that's that. Okay, this is cystic fibrosis. Are we clear about this? Sodium is neither hypo nor hyper. Okay, so the sodium level is maintained. The sodium level is maintained. Okay. Are we clear about this? Okay. Okay. Another thing is, when we talked about the X-linked recessive disorders, we studied about the diseases, but we didn't study about X inactivation. Okay, so what is X inactivation? X inactivation is lyonization, meaning that one copy of the female X chromosome forms a transcriptionally inactive bar body. So if the females have two copies, then one copy of the X chromosome is not responsible for transcription, and this results in the formation of an inactive bar body. So next time, if anyone asks you what is a bar body, can you say that it's a transcriptionally inactive X chromosome? Yes or no? Yes, this is high yield for NBME. Oh, it's a URL question too? Okay, I know it's a high yield NBME question. Okay. Female carrier is variably affected depending on the pattern of inactivation of the X chromosome, carrying the mutant versus the normal gene, right? For example, females with Turner syndrome are more likely to have X-linked recessive disorders. Okay, this has no link to bar bodies, but bar bodies are something which I want to discuss. Bar body is, especially if the females have two X chromosomes and one X chromosome is not working. As a result, if one X chromosome is not working, and if another chromosome, that is the X, another X is also not working, those females have a high possibility of developing diseases. But if there's one transcriptionally inactive X chromosome, the name that is provided for those X chromosomes is known as bar body. Okay. Can you guys name some diseases where we can find bar bodies? Where do we find bar bodies? Okay. Klinefelter syndrome. Very good. Klinefelter syndrome, Turner syndrome. It's also there. Okay. Okay, good. Having said that, can we move on to muscular dystrophies? Okay. So what is muscular dystrophy? These are very high yield. Duchenne, Becker's, and myotonic dystrophy. Okay. Duchenne's muscular dystrophy. Okay, what is Duchenne's muscular dystrophy? What type of mode of inheritance is Duchenne muscular dystrophy? What type of inheritance is Duchenne's muscular? Okay, it's an X-linked disorder which results in frame shift mutation. Do you guys remember when we talked about frame shift mutation and nonsense mutation? We talked about the muscular dystrophies as an example, right? So there's a frame shift mutation which results in, okay, you know what? Let me talk about this over here first, and then we can come here. Let me talk about this on a blank page. 1 second. Okay. First of all, so we're talking about Duchenne muscular dystrophy. First of all, it's an X-linked recessive disorder. What is the genetic defect? The genetic defect is frame shift or nonsense mutation. Nonsense mutation. Okay. As a result, we know that if there's a genetic problem, should there be a problem with the protein? Yes or no? Should there be a problem with a protein? all the time. If there's a genetic problem, there is a protein problem. We all know this, right? If there's a genetic problem, there's a protein problem. If there is a protein problem, then the protein that is affected over here is known as dystrophin. Dystrophin. Dystrophin is basically this is the largest protein coding human gene. The human gene codes for the largest protein, that is dystrophin. Okay, so what happens is there is loss of dystrophin. Now, what happens if there is loss of dystrophin? What is the function of dystrophin? Dystrophin is responsible for anchoring dystrophin. What it does normally dystrophin, what this does, normally dystrophin is responsible for anchoring muscle fibers. Anchoring the muscle fibers. Dystrophin is responsible for anchoring the muscle fibers, and this works in the skeletal muscle and the cardiac muscle. So what happens is it is responsible, when we talk about anchoring the muscle fibers, what we mean is that they're responsible for connecting the actin to, they are responsible for connecting the actin to two things, that is alpha and beta dextroglycan. Alpha and beta dextroglycan to extracellular matrix, to the extracellular matrix. So if I tell you that you have loss of this protein over here, will there be normal anchoring between the muscle fibers? Yes or no? Fast answers, please. Will there be normal anchoring between the fibers? Yes or no? Fast answers. Okay. As a result, can actin be properly connected to the extracellular matrix? No. If actin is not connected to the extracellular matrix, will there be proper muscular contractions? If there is no proper muscular contraction, can there be microscopic atrophy and myonecrosis as a result of loss of dystrophin gene? Yes. So what happens is these patients, they have microscopic myonecrosis. So they have myonecrosis. The muscle dies down. There is death of the muscle. There is death of the muscle. As a result, what happens is when there is death of the muscle, this dead muscle, what happens is that this dead muscle, especially this dead muscle over here, is replaced by fat. Okay, in pathology, this dead muscle is replaced by fat. As a result, what happens is in the calf, you see a hypertrophy, which is a pseudo hypertrophy. Okay. How many people do we have over here who hits leg days, who loves leg days? Do you guys love leg days? Do we have any student who works out at the gym and goes for leg days? Anyone? Leg days. Okay. What happens when you hit your calf? When you hit your calf muscle, do you have hypertrophy or pseudo hypertrophy? Well, what happens when you hit your calf muscle? Do you have hypertrophy or pseudo hypertrophy? Hypertrophy, meaning that when you work out on your calf muscle, you make sure that the muscles grow. But over here, what happens is in these patients, they look like they have been working out, they have been hitting leg days every day because their calves are huge calves. But the thing is, it's pseudo hypertrophy because it looks like a hypertrophied muscle, but it's actually the muscle fibers replaced by fibrofatty tissues. That's why this is called pseudo hypertrophy. And this sign in a young child who with pseudo hypertrophy, this sign is known as, who knows the name of the sign? Gower sign. Okay, this is Gower's sign. This is Gower's sign. Okay, that's that. Are we all clear about the pathology of Duchenne's muscular dystrophy? Yes or no? Okay, good. Okay, before we return to our text, there is another type of muscular dystrophy which I want to talk about. This muscular dystrophy is known as Becker's. This is known as Becker muscular dystrophy. Becker's muscular dystrophy. Becker's muscular dystrophy is basically, it's what type of a disorder is Becker's muscular dystrophy? Fast answers, please. X-linked recessive. Once again, it is also an X-linked recessive disorder. Over here, you also have deletion of the dystrophin gene, but what happens is that a deletion is a non-frame shift. It's a non-frame shifted deletion. It's a non-frame shifted deletion. If it's a non-frame shifted deletions, can we assume that the Becker's muscular dystrophy is less severe than the Duchenne's muscular dystrophy? Yes. So Becker's is basically, it's more, this is more or less the same as Duchenne muscular dystrophy, but it is less severe than the Duchenne muscular dystrophy. Okay, so that's that. The only thing you have to remember from the Becker's muscular dystrophy is this is also a deletion of the dystrophin gene, but it's a non-frame shift deletion. Okay, it's a non-frame shift deletion. Now let's return to our text. Let's see what our text is. Okay, so Duchenne's muscular dystrophy is an X-linked disorder, typically due to frame shift deletions or nonsense mutation. Put a star over here. This is high yield. Truncated or absent dystrophin protein. This is for your NBME. What type of protein is lost? And the answer is dystrophin gene. Progressive myofiber damage. Weakness begins in pelvic muscles and progresses superiorly. So if the patients will complain that you will have a patient who will come to you with weakness in the lower part of the body, and the weakness will be progressively moving upwards. So will you receive a question about the patients with the weakness in the arms or in the leg muscles first? Fast answers, please. Weakness in the arms or the leg muscles first. Very good. The weakness will be especially in the legs area, right? As a result, what happens is we talked about how there's myonecrosis. As a result, there's fibrofatty replacement. And what happens is these patients, they have pseudo hypertrophy of the calf muscles. And this results in waddling gait. Obviously, if the calf muscles are so huge in a little baby, it will be difficult for them to walk. So what are the clinical features of Duchenne's muscular dystrophy? First of all, is muscle weakness, especially in the legs and then to the arms. And next one is abnormal gait. Next one is abnormal gait. And obviously you have Gower sign, which is also positive. Gower sign, the patient uses the upper extremities to help stand up. This is classically seen in Duchenne muscular dystrophy, but it is also seen in other muscular dystrophies and inflammatory myopathies. So along with the fibrofatty replacement of the pseudo hypertrophy, these patients, what they will do is, since they cannot use their pseudo hypertrophy leg to stand up, what they would do is these patients, in order to stand up, they would need the help of some sort of an object to hold on to, and they will stand up using the upper extremities. So this is known as Gower sign. So Gower sign is not only the pseudo hypertrophy, it's also the sign of the patient standing up by using something. So that's that. Okay, and this is extremely high yield because you will receive a question, especially on this one, that USMLE will use this piece of information to see whether you can identify the diagnosis that is Duchenne muscular dystrophy. Okay, so that's that. Next one is Duchenne is basically, the gene is dystrophin gene, which is the largest protein coding human gene. This gene is the largest gene which codes for the protein. That is, the protein which this gene codes for is the largest. And since this is so large, the chance of mutation is very high. It makes sense because if there are millions of nucleotides which are responsible for coding the dystrophin protein, right? Then any one of the nucleotides can undergo mutations, and this can result in nonsense or frame shift deletion. So since it's the largest protein, this is the most common mutation that is possible. Dystrophin helps anchoring muscles. We talked about this before, how it is responsible for connecting the intracellular actin to the alpha and beta dystroglycan, which are connected to the extracellular matrix. As a result, if you have loss of the dystrophin gene, this connection is lost. If this connection is lost, there is decreased muscular contraction, resulting in microscopic atrophy and myonecrosis. Okay, myonecrosis. How do you diagnose a patient with Duchenne's muscular dystrophy? Obviously, do you guys realize that if you have muscular breakdown, then the creatine kinase will increase? Yes or no? Creatine kinase will increase. Increase in creatine kinase will happen. Along with this, there will also be increase in aldolase. Increase in aldolase, but high CK is more important. Out of CK and aldolase, high CK is more important. And obviously, you can also do a genetic test. If you do a genetic test, then you can see there is absence of the dystrophin gene. And this is all you need to know about Duchenne's muscular dystrophy. One more thing over here, obviously, is the picture of the histology. That as you can see there, these are fat cells inside the muscle fibers. So if you take a small piece of muscle from the calf and you study this under the microscopy, you can see that there's fibrofatty replacement of the muscle tissues, and this is pseudo hypertrophy. Okay, are we clear about this? Okay, good. Next one is Becker's muscular dystrophy. Becker's muscular dystrophy. It's an X-linked recessive disorder, typically due to a non-frame shift deletion in dystrophin gene. Okay, so what happens is it is less severe than Duchenne's, and the deletions, they can cause both Duchenne's and Becker's, but the Becker's muscular dystrophy, 2/3 of the cases have large deletions spanning one or more exons. Do you guys remember we talked about the fact how in order to produce a mature mRNA, you have to get rid of the introns and you have to get rid of the introns, right? Yes or no? You have to get rid of the introns, right? So if there is a very massive mutation in one or more of the exons, do you guys remember how the exons are responsible for initiating transcription? Okay. If I were to tell you that there were mutations in any one of those exons, that is what I want to mean about the pathology of Becker's muscular dystrophy. This is not very high yield. This is not something which you really have to know. All you have to know about Becker's muscular dystrophy is it's a non-frame shift deletion. That is very widely asked over here, non-frame shift deletion. And Duchenne is a frame shift deletion. Okay, are we clear about this? Yes or no? Okay, good. Now, okay. Now we are going to talk about another disease. This disease is known as myotonic dystrophy. Okay, this disease is known as myotonic dystrophy. Before I talk about the myotonic dystrophy, I want to share a picture mnemonic from myotonic dystrophy, because the findings over here are a bit difficult to understand, and I don't want you guys to blindly memorize the findings. Do you want us to begin with the picture mnemonics of myotonic dystrophy so that it's easy for us to recall? Yes or no? Okay. Okay, 1 second. Okay, myotonic dystrophy. Once again, can we have, is Dr. Jordan still here in our lecture? Dr. Jordan, are you still here? Okay. Would you be kind enough to take the responsibility once again? Thank you so much. Okay. Thank you so much. Okay, so let's begin with this. Myotonic dystrophy.
Welcome to section 9.2 of genetics. In this section, we will be memorizing the details of myotonic dystrophy. So let's get started. This scene takes place at a dump, as indicated by the sign here. Deep in this chasm is where people put garbage. Dump will help you remember that the DMPK gene is affected in myotonic dystrophy. So dump or dump K gene. Our story is like a gritty crime movie where mobsters are trying to dispose of a dead body. Look how emaciated this body is. This wasted body will help you remember muscle wasting in myotonic dystrophy. Like most mobster movies, the cops show up and create a climactic showdown, and our story is no exception. Now notice the dominoes hanging from the rear view mirror of the cop car. Dice are more often placed in rear view mirrors like this, but dominoes fit the story better. In any case, these dominoes will help you remember that myotonic dystrophy is an autosomal dominant disorder. Quickly, a cop jumps out of the vehicle. In his excitement, he repeats on his communication system, "We caught these guys. We caught these guys." So caught these guys. If you look at the first letter of each word in his proud declaration, caught these guys, you will see CTG or CTG repeats. Now it's these CTG repeats within the DMPK gene that cause all the problems. If you can remember that in our story, the cops caught these guys, you will remember CTG repeats in myotonic dystrophy. Now, while the cops weren't looking, an obnoxious teenager tagged the side of the police car with his initials, MPK. MPK stands for myotonin protein kinase. As indicated by the name, myotonin protein kinase, this MPK is a protein, and it's overexpressed from all these CTG repeats in the DMPK gene. Now notice the lights beaming at these two mobsters in the back. You can see this guy's eyes are reflecting the light in a weird way. This is to help you remember cataracts. Patients with cataracts often complain of having a severe glare from car headlights at night. Often this complaint alone is what leads patients to the doctor. So light reflecting from eyes, cataracts. Now look at the lights beaming off the forehead of this guy. The reflection of light off of his shiny bald head will help you remember frontal bossing. Frontal bossing refers to balding of the frontal portion of the patient. So patients with myotonic dystrophy often have frontal bossing. So light reflecting off bald head, frontal bossing. As you'd expect, these mobsters have more than just one victim. Look at this poor guy. He's getting zapped by the car battery behind him. Pretty cruel. Anyways, it's causing all of his muscles to contract and spasm uncontrollably. Look how stiff he looks. His legs sticking out, his legs straight, arms to his side. He's completely contracted. Now this will help you remember that patients with myotonic dystrophy have myotonia, which means they have a hard time relaxing their muscles, just like this guy. For example, a patient with myotonic dystrophy may grip your hand in a handshake, but they won't be able to readily let go. This is myotonia. So this victim is tensed up from the electricity coming from the car battery, which refers to myotonia. Fortunately, this poor myotonic man tensed his leg in just the right way to get some poetic justice for this mobster. You can almost see his testicles explode with this myotonic kick. This testicular destruction here will help you remember testicular atrophy, which is common in patients with myotonic dystrophy. Now let's look more closely at the victim's shirt. Can you see the little heart? The fact that the electricity seems to go directly to his heart indicates that patients with myotonic dystrophy often have arrhythmia. So shocking the heart with electricity for arrhythmia. And with the arrhythmia of the myotonic man, we have wrapped up all the details you need to know about myotonic dystrophy.
Okay. Dr. Georgian, could you post or take a picture of this one? Were you able to take a picture of this one? Yes or no? Okay, good. Thank you so much. So how was the small cartoon information? Was it good? Yes or no? Okay. Now let's talk about myotonic dystrophy. Okay, now let's talk about myotonic dystrophy. First of all, myotonic dystrophy. What type of a disorder is myotonic dystrophy? What type of disorder is this? The mode of inheritance? Okay, good. Now, okay, it's a autosomal dominant disorder. What happens over here is that you have a trinucleotide repeat disorder. If there's a trinucleotide repeat disorder, what type of a genetic term would you use to identify this disease? Which genetic term will you use to identify this disease? Anticipation. Very good. Very good. Over here, so the repeat disorders, what is the trinucleotide repeats? Which nucleotides are repeated over here? CTG. CTG, and what was the mnemonic which you just saw? Caught these guys, right? We caught these guys, so CTG. CTG, nucleotide repeat disorders. What is the name of the gene that we just learned? The name of the gene that we just learned from over here? DMPK. DMPKG. As a result, you have CTG trinucleotide repeats in DMPKG. If you do a DNA amplification test and then you do a chromosomal electrophoresis or a DNA electrophoresis, can you assume that in the DMPK chromosome, there could be amplification of the layers like this because there is repeat disorders? Yes or no? Yes. This is a high yield question from NBME. Okay. They will tell you that in DNA electrophoresis, there is DNA amplification at the region of DMPK gene, and the diagnosis is myotonic dystrophy. Okay, myotonic dystrophy. Now, let's see if you guys can name all the clinical features which you have just learned from the cartoon. What are the clinical features? Fast answers, please. Clinical features: muscle wasting, then arrhythmia, then cataracts, then balding, then frontal bossing, okay, then myotonia, the most important one, okay. So are we confident about myotonic dystrophy? Myotonic dystrophy. Okay. The muscles in myotonic dystrophy, will they be rigid or not rigid? Rigid. Did you guys mention testicular atrophy? Testicular atrophy. Okay. Okay. So if you receive questions from myotonic dystrophy, will you be confident or not? Okay, good. So let's see the text. Let's see if we have so-so. Okay. So what do you mean by so-so? So what is the percentage of your confidence? Above 60% or less than 60%? Semi-confident. Okay. How do you become extremely confident from semi-confident? What is the best way to become? This question goes out to everyone. Let's see who can provide the right answer. How to become confident from semi-confident? Questions, UWorld. Okay. So, Dr. Sabira, if you want to become extremely confident from a so-so level of confidence, what do you have to do? Okay, good. Okay, hopefully every one of you will do very well. So that's that. Let's talk about myotonic dystrophy. It's autosomal dominant CTG trinucleotide repeat expansion in DMPK gene, abnormal expression of myotonin protein kinase resulting in myotonia, muscle wasting, cataracts, testicular atrophy, frontal balding, and arrhythmia. That's that. Okay. Now we are over here to a very rare and extremely difficult to diagnose sort of a disease. This disease is known as Rett syndrome. I remember Rett syndrome because I received this question in my own USMLE Step 1 exam. When I sat for my exam, a long time back, obviously, then I remember coming across this question, that is the Rett syndrome. Okay, so although it's a pretty rare disease, I am going to provide this with high yield stars because this comes in actual exams. Okay, how do you remember all of these diseases? What do we do with Rett syndrome? What do we do with Rett syndrome? How to remember Rett syndrome? Rett syndrome. Physio. Very good. Good. Okay. I hope you guys do not get annoyed by my rhetorical questions. Okay, I have a habit of asking rhetorical questions, so please forgive me and do not get annoyed by this. It's a bad habit. I'm trying to overcome this. I keep on asking rhetorical questions, the answers to which I know myself. Okay, Rett syndrome. Rett syndrome, okay. So is Dr. Jordan ready? Dr. Jordan, are you ready?
Welcome to section 7.
Okay, so let's begin with Rett syndrome.
Point 2 of genetics. In this section, we will be memorizing the details of Rett syndrome. Let's get started. This story takes place on a street between a retro diner and a mechanic shop. The retro diner should help you remember that this is Rett syndrome. Now, all of the chaos started when this taxi driver reversed out of the mechanic shop in a great hurry. You can even see the reverse track marks all over the road. This reversing will help you remember that in Rett syndrome, patients experience a regression of abilities. We will get into the ways in which Rett syndrome patients regress in a moment. Just know that these patients develop appropriately for a while and then regress and develop issues. In his careless exit, this driver knocked over 2 streetlights, causing them to fall in this X position. This X that is formed by the lights will help you remember that Rett syndrome is X-linked. Also notice that one of the lights smashed down onto this table back here. This nice couple was playing dominoes outside this cafe. You can see dominoes falling everywhere. This is to help remind you that this is X-linked dominant. Now notice these little girls running away from the falling streetlight. Thankfully, they made it out of the way in time. But notice that they are girls. This is to remind you that only females are impacted by Rett syndrome. This is because Rett syndrome is X-linked dominant. Boys only have 1 X chromosome, so an altered X chromosome is devastating, causing them to die in utero or shortly after birth. As you may know, most food establishments have a happy hour in the early afternoon when things are slow. During their happy hour, restaurants will give out cheap drinks or other items at very cheap prices. Now this retro diner is no exception. On the window, you will see happy hour runs from 1 to 4. This happy hour time frame will help you remember that symptoms begin between ages 1 to 4. Remember that this is regression, so they will develop normally until the disease starts to manifest itself, at which point regression begins. This light post smashed a poor little drive-through clown. Look how squashed he is. Man, it's really fortunate that those girls got out of the way. This is to help you remember that Rett syndrome causes growth failure. Now this clown is normally how the drive-through customers place their order. Once the clown got all smashed up, the voice box got destroyed. You can see the garbled sounds coming out of it. This clown's garbled speech will help you remember verbal regression in Rett syndrome. Do you remember how it was a taxi that caused all of this chaos? Silly taxi driver. Remembering this taxi will help you recall that Rett syndrome patients have ataxia. So taxi, ataxia. Now the careless driver knocked over a big motor in the mechanic shop, causing it to crash to the ground. This motor will help you remember motor regression in Rett syndrome, so their ability to move appropriately is impacted. That makes sense. They have ataxia, like we just discussed. Notice how the motor is still spinning and rattling around. It's kind of like it's having a seizure. This seizing motor will help you remember that these patients are prone to seizures. The mechanics in this shop are super busy. So busy, in fact, that they don't have time to go to the bathroom. So they just pee in this gross bucket labeled mechanic pee. This nasty mechanic pee bucket will help you remember that in Rett syndrome, the affected gene is MECP2. Mechanics P2, after all. So mechanic P, MECP2 gene. This mechanic, hard at work, is trying to wash off all the oil from his hands. Oil is really hard to get off, so he's really wringing his hands. This hand wringing will help you remember the stereotyped hand wringing seen in Rett syndrome patients. And with stereotyped hand wringing, we have completed our story on Rett syndrome.
Okay. So this is Rett syndrome. Okay, so let's talk about Rett syndrome in a blank page. Did we understand the picture mnemonics? Yes or no? Did we understand the picture mnemonic? Guys, did we understand the picture mnemonic? Okay, now let's talk about the disease. Dr. Jordan, were you successful in taking the picture or the screenshot of Rett syndrome? Okay, good. Okay, it was difficult for me. Do not worry. Now I'm going to break it down. Okay. Okay. So what is Rett syndrome? First of all, Rett syndrome is a disease where you have, okay, let me start with the UL question. The question is, you have a young female patient who is 3-year-old. The parents come to you and tell you that their child was developing normally. All of a sudden, her developments stopped and she learned how to stand up and walk, but right now she cannot walk at all. She learned a few words, but right now she cannot. say those words anymore. Okay, and she could stack up toys on top of another, but right now, all of a sudden, her abilities to stack up toys has failed. Along with this, the child also has ataxic gait and some sort of an abnormal movement of the hand. Okay, so young female child, so young female, age is usually 1 to 4 with regression. What do you mean by regression? Regression means to decrease in amount, right? So what are the regressions? The regressions are motor regression, verbal regression, and cognitive regression. Cognitive regressions. These three are the regressions as that will be complained by the parents to you as a physician. Okay, and you will notice that this child has ataxia, seizures, and growth failure. If you see any type of these findings in a patient in an age of 1 to 4 year old, the disease I want you to think about is Rett syndrome. Rett syndrome. What type of inheritance is Rett syndrome? Rett syndrome. What type of inheritance is Rett syndrome? It's an X-linked dominant. Is it a dominant or a recessive disease? It's a X-linked dominant. So how many X-linked dominants did we learn today? Fragile X, then hypophosphatemic rickets, Alport syndrome, and Rett syndrome. Okay, good. So it's an X-linked dominant disease. X-linked dominant disease. And due to this X-linked dominant disease, what is the mutation on which gene do we have the mutation? On which gene do we have the mutation? The mutation is in MECP2. MECP2. Okay. Another thing is this disease is a sporadic disease, meaning that it can affect either due to inheritance or it can happen out of the normal ordinary or unordinary, meaning it's sporadic, that there's a possibility it can happen even without inheritance. That's what we mean by sporadic. This is Rett syndrome. Can we remember the findings of Rett syndrome from the picture mnemonic, which we have just discussed? Yes or no? Okay. I wanted to use the picture mnemonic, especially for the genes, especially for the gene MECP, for mechanical P, MECP, MECP. Okay. This is high yield. This is high yield. Let's see what our text says. Our text says that Rett syndrome, it's a sporadic disease. Okay, in Rett syndrome, it's a sporadic disease. It's almost exclusively seen in girls. Affected males die in utero or shortly after birth. Most cases are due to de novo mutations of MECP2, high yield. If they can mention MECP2 in the questions directly, and if they do, they are directing you to Rett syndrome. Symptoms of Rett syndrome usually appears between age 1 to 4 and are characterized by regression. The regressions are in motor, verbal, and cognitive abilities. As a result, these patients have ataxia, seizures, and growth failure, and stereotypical hand-wringing movement. Hand-wringing movement. Okay, are we clear about this? Yes or no? Okay. Here, 1 second. I want to show you guys one thing. Let me show what I mean by the hand wringing. This is the hand wringing movement. They keep on moving their hands like this. Okay. Children's, especially females, age 1 to 4, who keeps on moving their hands like this. If you see any patient like this in your clinic, then think about Rett syndrome. Rett syndrome. Okay. Good. So that's that. Okay. Now we will talk about Fragile X syndrome. Okay. Everyone, are we clear about Rett syndrome? Are we clear about Rett syndrome? Yes. Okay, good. So can we start with the Fragile X syndrome? Okay, good. What are we going to do with Fragile X syndrome? We have a lot of informations over here. What we are going to do is learn another picture mnemonic. Okay, so thank you once again to Dr. Jordan for taking the responsibility of taking so many pictures for us today. Okay, hopefully you guys will have access to this pictures in the discussion group, which Dr. Jordan have already posted there. Okay, and the next time when you want to do a revision, do you guys want to use the picture mnemonic to do the revision? Yes or no? Yes. Okay. Make sure you guys use the picture mnemonics, or else you will forget about
Welcome
this.
to section 9.
Okay. Make sure you guys use the picture mnemonics to do the revision if you have to, or else you will forget about this. Okay. Okay. Let's begin.
9.3 of genetics. In this section, we will be memorizing the details of Fragile X syndrome. So let's get started. This story takes place in a boxing ring. As you can see, there is a big crowd here to see the fight. But who are they here to see? A boxer named Gennady Golovkin. In fact, he is the champion of our match. As you can see with this banner displayed here. Champion Gennady Golovkin. As you may already know, Fragile X syndrome is a trinucleotide repeat disorder. The trinucleotide that gets repeated is CGG, Champion Gennady Golovkin. If you remember that the champion of this image is Gennady Golovkin, you will remember CGG repeats cause Fragile X. Now, just to help reinforce that Gennady Golovkin is an actual person, I've included this image. And here is another picture of him. And he's such a good boxer that he was even placed on the stamp of Kazakhstan. Now let's get back to our fight and bring him in. Let's bring in his opponent. Bam. Look at Gennady knock out his opponent. Look at him fall. His arms spread apart and even his legs. Clearly, he is going down for the count. Notice that his arms and legs form an X, as in the X chromosome in Fragile X. The reality is that this boxer was quite fragile, since Gennady only needed to hit him once to knock him out. Now look at his jaw, which gets long and unhinged from the knockout punch. This is to remind you that people with Fragile X have a long, prominent jaw. A knockout punch like this one has the potential to rile up a crowd, and this punch is no exception. In fact, one man comes running up to the ring in anger over this knockout punch. This man is a farmer, and he actually bet his farm against Gennady Golovkin. Naturally, Gennady's victory upset him. Anyways, this farmer will help you remember that it is the FMR1 gene that is the problem in Fragile X. And as you can see, the farmer is being bound by these guards. These guards are so good at their jobs that they have effectively prevented the farmer from jumping into the ring and fighting Gennady Golovkin himself. In this way, the guards represent methylation of the FMR1 gene, effectively inactivating the gene. So again, guards binding the farmer represents methylation and inactivation of the FMR1 gene, leading to the symptoms of Fragile X. So let's talk about some more of the symptoms. Another member of the crowd joins the farmer in this ruckus. Maybe he knows the farmer, or at the very least, he sympathizes with his loss. So he comes and pulls on the ears of one of the guards. Notice that he is pulling them up and out. This will remind you that patients with Fragile X syndrome have large everted ears. Now here's an image of a child with Fragile X syndrome, and his ears demonstrate the typical phenotype seen in Fragile X patients. In the background, outside the ring, notice two boxers preparing for the next fight by beating a punching bag. Notice what this bag looks like? It kind of looks like a giant set of testicles. Thankfully, this will help you remember that people with Fragile X syndrome often have enlarged testicles. And since Gennady has already won the match, he is being awarded an MVP belt. This will help you remember that people with Fragile X syndrome often have mitral valve prolapse.
Okay. So this is everything you need to know about Fragile X syndrome. Dr. Jordan, could you take a picture of this? Yes or no? Okay, good. So let's talk about Fragile X syndrome. Fragile X syndrome Okay, Fragile X syndrome is basically is it a trinucleotide repeat disorder? Is this a trinucleotide repeat disorder? Yes or no? Yes. The trinucleotide repeat disorder, what is the name of the gene in which there is trinucleotide repeats? So the repeat is CGG. CGG. Okay. What is the name of the gene? The name of the gene is FMR1. FMR1. Okay. As a result, if there's a trinucleotide repeat disorder in FMR1 gene, what happens is that over here in this gene, you have hypermethylation. Hypermethylation. If I tell you about hypermethylation, can we talk about the fact that there is decreased genetic expression of the gene? Yes or no? Yes. So as a result, what happens is there's hypermethylation and there is decreased genetic expression, due to hypermethylation. Okay. Okay. Now, what are the findings or clinical features of Fragile X? Let me see if you guys can remember everything which you just saw. Large ears, then mitral valve prolapse, then long jaw, and macro and what? Macro orchidism or enlarged testis. Macroorchidism. Okay. Large ears, mitral valve prolapse, long jaw, macroorchidism. Do you guys remember seeing the picture of the small baby with the Fragile X syndrome? Of the little baby with Fragile X syndrome? Yes. So that was Fragile X syndrome. Okay, let's look at our text to see if we have learned everything. Okay, so Fragile X syndrome is an X-linked dominant inheritance. Okay, so how many? Once again, Fragile X, Alport syndrome, hypophosphatemic rickets, and Rett syndrome. These are all X-linked dominant disorders. Trinucleotide repeat disorder in FMR1, resulting in hypermethylation and decrease in genetic expression. Most common inherited cause of intellectual disability. Down syndrome is the most common genetic cause, but most cases occur sporadically. The most common genetic disorder is Down syndrome, and then after that, you have Fragile X syndrome. Findings are post-pubertal macroorchidism, long face with large jaw, large ears. Autism is also there to some extent. Mitral valve prolapse and hypermobile joints. Out of these, large jaw, large ears, and macroorchidism, these are high yield. These are extremely high yield. Okay, and obviously I cannot stretch how important CGG is. CGG, and the gene is FMR1. Okay. Myotonic dystrophy. What is the nucleotide repeat disorder? Myotonic dystrophy. What is the nucleotide repeat disorder? CTG. What is the gene in myotonic dystrophy? Name of the gene. DMPK. What is the trinucleotide repeat disorder in Fragile X syndrome? CGG. What is the name of the gene? FMR1. What are the findings of myotonic dystrophy? Clinical findings. What are the clinical findings of myotonic dystrophy? FMR1, cataract. What else? Myotonia, balding, arrhythmia. Okay. What are the findings of Rett syndrome? Rett syndrome. Seizures, regression, ataxia. Okay, good. What is the age for Rett syndrome? 1 to 4. Who are more affected, males or females? Females. Okay, so you guys sound really confident to me. How do you guys feel about your recalling capabilities of the text which you just learned? Confident. Okay, good. Okay. Okay. Right. Very good. Okay. So far we have studied about Fragile X. We have studied about myotonic dystrophy. We have not studied about Huntington's and Friedreich's ataxia. This piece of information that we have from this page, can we study this after we are done studying Huntington's and Friedreich's ataxia? This piece of information, because I want to talk about these things after we are done studying with this one. Okay. Okay. Now let's move on to autosomal trisomies. Okay. Are we all ready to autosomal trisomies? Everyone? Okay. Can we take a quick break? Okay. How long do you guys want to take the break for? How long do you guys want to take the break for? 10 minutes? 5 minutes? 1 minute? 1 minute is not a break, Dr. Hussam. Okay. 5 minutes to 10 minutes. Okay. So let's give you guys 10 minutes. 5 to 10 minutes. Okay. I really want to be done with these 2 pages so that we can start fresh with nutrition from tomorrow. Can you show us your plan for biochemistry? What plan are you talking about, Dr. Iman? The lesson plan? Are you talking about the lesson plan? How many days? I'm not, to be honest, it's extremely difficult for me to tell you how many days with biochemistry, because do you guys want, because to be honest, I can finish biochemistry in 1 week, but what that will do is that will not allow us to understand how, that will not allow us to understand if every one of us are learning or not. You understand what I mean? So that's why it's extremely difficult to say before if we can finish biochemistry. Next one. Do you think we will finish first aid before February 1st? We want to finish first aid as soon as possible. So that's why, to be honest, I'm trying to do my best every day to make sure that we finish it in that time span of 3 to 4 months. Okay. So we are trying our best, but if we can't finish, so what happens is, do you guys want me to rush through the last topics of basic science, or should we should we take things at this pace, because it's important to what happens is no, my apologies. I'm asking for a feedback, because I'm going to tell you I'm asking for a feedback, because if you guys think that I'm taking a lot of time, then I can actually speed things up, because I can finish these things very quickly. Okay, but the best thing is these things now, since we are watching physio, do you guys realize that since we are taking just a little bit more time, not a lot, we are taking just a little bit more time than what we said. Do you guys realize that if you really want to remember the text in first aid right now, it's better? For example, you can easily recall the text which you have learned yesterday, and this really shows in terms of your UWorld questions. I was asking the students earlier this morning how many questions they got right from UWorld, and most of them said that they did the questions right exceptionally well in the ranges of 80% or more, and that is very, very good. So I guess if this is the way that's what's working, if we have to take just a little bit more time, it's good for everyone, more or less, because biochemistry and microbiology, and these are the things which are a bit on the complicated side. Although we should be really glad that we are done with the organ systems. But the thing is, if we are done with microbiology after biochemistry, then most of our first aid will be done. After that, we'll just need immunology and that. Okay. So hopefully we'll try to finish it off by the 1st of Feb or mid of Feb, but we'll do our best. Okay. Okay. So if you guys have any feedbacks for us, please send us an email and let us know, and we'll try to work around it. Okay. We had a lot of feedbacks from our students in the past, and we try to stick to them as firmly as possible. So we're always open to feedbacks. Okay. So with that being said, let's not waste any time and take that break. Okay. The reason I asked is I want to incorporate more pathoma into my schedule. Okay. If you want to incorporate more pathomas, then you can watch 1 hour of Pathoma every day. To be honest, that's actually that's actually good enough. You understand what I mean? There, because the Pathoma videos, if you can watch 1 hour every day, that's more or less enough. Okay. Okay, so let's take that break and let's come back and finish the last 2 pages. Okay, is anyone back from their break? Is anyone there? Can we begin with the lectures? Okay, do you guys have any plans for your New Year's that's coming up ahead? Do you guys have any plans for New Year's? Okay, good, very good. That makes all of you guys, and except me. Okay. You guys should be really careful with, because the second wave of coronavirus is also rising. So be really careful when you guys are going outside. Okay, good, very good. Did you guys have a good Christmas though? Okay. No? Why? Why didn't you have a good Christmas? Okay. Went to our cottage. Okay, good. That's very good. We will be having classes on the December 31st and the 1st of Jan. Will that be affecting you guys? Yes or no? Okay. Okay, because we already have to make up for a class which we missed out on Christmas. So we do not want to take any other break for New Year's. Is that okay? Okay. Okay. Did you guys get really confused when we took that break in Christmas out of nowhere, and when you guys didn't receive the URL in the morning? Okay, but it was like a small Christmas miracle, wasn't it? And okay, it was like a very small Christmas miracle. The class gets canceled and it's a holiday, and you guys got to enjoy 3 days of your weekends. Okay, good. Okay, so let's begin with autosomal trisomies. Okay, let's begin with autosomal trisomies. Autosomal trisomies, these are extremely important: Down syndrome, Edwards syndrome, and Patau syndrome. Okay, and once again, the best way to do these and to remember all this information over here is we're going to go text by text, of course, but can we go through physio to see if we can use their pictures to remember all the findings in the first aid? Yes or no? Okay. So we're going to use physio to learn about all the information about Down syndrome. And then we're going to take it from there. Are you guys on board? Is everyone here? Is everyone back from their break? Can we begin? Okay. Okay, let's begin. Dr. Jordan, are you ready for the screenshot? Okay, good. Thank you. Once again, thank you, Dr. Jordan, for taking the responsibility. Let's do this.
Section 10.1 of genetics. In this section, we will be memorizing the details of Down syndrome. Let's get started. This scene takes place in an unassuming field between a river and the home of an innocent family. However, as you will soon see, this location is currently the site of some serious action. To emphasize my point, look at this guy, clearly falling down to the ground. Down for Down syndrome. But why is he falling down? And where is he falling from? This will all make sense at the end of the story. It all started with a simple robbery. See this mugger holding a gun at this innocent man? The man has his hands up, you know, like you do when you have a gun pointed at you. Now take a close look at his palm. Notice that top crease, the transverse crease? He only has one. Most people have two transverse palmar creases. Anyways, Down syndrome patients often have a single transverse crease on their palms. So this victim with a single prominent palmar crease will help you remember the single palmar crease in Down syndrome. Now, unfortunately, this single palmar crease guy is not the only victim of this stick-up. Notice this nice old lady to the left? She is holding her newborn twins. The fact that she is old will help you remember that the risk of Down syndrome increases with maternal age. Additionally, Mothers that conceive at older ages are at an increased risk of non-disjunction occurring during meiosis. If non-disjunction of chromosome 21 occurs, then this leads to Down syndrome. If you look closely, you will see that these twins are conjoined, helping to remind you of non-disjunction. So this old lady with conjoined twins for non-disjunction occurring with advanced maternal age. Now, of course, this sweet old lady didn't conceive these children all by herself, so we also included her elderly husband in this story. Unfortunately, because her husband has Alzheimer's dementia, he's not much help in this situation. As you can see, he's just yelling at his candy bar, completely misunderstanding the crime right in front of him. This confused old man will help you remember that Alzheimer's dementia often occurs in Down syndrome patients, even at earlier ages than typical people. Now notice the candy bar. It's a Hershey's chocolate bar. This will help you remember that patients with Down syndrome often have Hirschsprung's disease. So Hershey's candy bar for Hirschsprung's disease. Fortunately, the family's dog is putting up a fight. Notice how this dog protects his puppies by biting between the big and small toe of the assailant. This will help you remember that patients with Down syndrome often have a wide space between the big toe and the adjacent second toe. So biting between the first two toes stands for increased space between the first two toes. Now the puppy's mother is trying to carry them away from the fray. And as dogs do, she picks them up by gently biting the nape of their neck, or the nuchal region. You can see this nuchal skin on this puppy all stretched out. This will help you remember that fetuses with Down syndrome often have nuchal translucency, meaning that fetal ultrasounds will reveal a translucent nuchal region, making it look bigger and stretched out, kind of like this dog. Stretched out nuchal region on this puppy stands for nuchal translucency on fetal ultrasound. And scared of the ferocious dog biting his friend, another mugger panics and trips. Thankfully for the family, he smashed and bent his double-barreled shotgun. Double kind of sounds like duodenum. So the double-barreled shotgun should help you remember the duodenum. And the fact that the double-barreled shotgun is bent up and totally blunted will help you remember that patients with Down syndrome often have duodenal atresia. So blunted double-barreled shotgun for atresia of the duodenum. Hoping to avoid getting caught by law enforcement authorities, another bandit is attempting to escape on an ATV. As you can see with the smoke bursting from under the hood, this ATV is not working. This will help you remember that patients with Down syndrome often have atrioventricular cardiac defects. So broken ATV for atrioventricular defects. Now the bad guys are trying to reach this river to escape. Notice that things didn't work out for this guy. As you can see, he's floating face down in the river, surrounded by his own blood. This bloody river will help you remember blood cell cancer, or leukemia, which is quite common in Down syndrome patients. Now there are two types of leukemia to remember: acute myelogenous leukemia and acute lymphoblastic leukemia. We have a memory hook for each one of these. Look at these mile markers along the river, and notice how each mile marker has a little pink ribbon tied around it. Pink ribbons such as these are often used to represent hope as it relates to cancer. So we like to use pink ribbons to represent cancer. And in the context of a bloody river, this should help you remember that we are dealing with a blood cancer, leukemia, specifically myelogenous leukemia. Remember myelogenous because of the mile markers. And patients with Down syndrome have an increased risk of acute myelogenous leukemia, or AML. So again, pink ribbons on mile markers for acute myelogenous leukemia. And the second type of cancer is acute lymphoblastic leukemia. And the lymph in lymphoblastic refers to white blood cells. So let's discuss this guy with the rocket launcher a little bit more. As you can see, this launcher has blasted a white rounded rocket at the helicopter. The round and white nature of the rocket resembles white blood cells, or lymph, as in acute lymphoblastic leukemia. So again, the white rocket blasting against the helicopter stands for acute lymphoblastic leukemia. And with the helicopter spinning out of control, we can now see why this guy was falling down. Remember that guy falling from the sky? Our Down syndrome guy? Well, he was trying to stop the bad guys. As you can see with his gun falling through the air and the dead body in the river, it looks like he was successful in taking out at least one of his targets. Now I want to turn your attention to this canister that has fallen out of the helicopter. As it hit the ground, it got smashed up on one side. This canister will help you remember epicanthal folds. And epicanthal folds are common in Down syndrome patients. So again, smashing canister for epicanthal folds. Now the helicopter has been using its spotlight to identify the perps rushing through this field toward the river. The fact that they are brushing through this field and getting spotted will help you remember brush-filled spots. Now brush-filled spots are interesting features of the iris in the eyes of Down syndrome patients. So again, these bad guys spotted rushing through the field stands for brush-filled spots. So now let's look back at this robber on the broken ATV. He must have crashed into a street sign before arriving. Maybe that's why the ATV is broken. And if you look closely, it looks like the street sign came from E Street. E Street kind of sounds like estrogen. And the fact that the sign is broken and on the ground indicates that estrogen is low in the maternal serum. So again, fallen E Street sign for low estrogen. Now look at this tree over here on the left. One of the robbers took the time to carve their first initial into the tree. Kind of a strange thing to do in a stick-up. I guess they must have done this before the helicopter arrived. Anyways, this poor tree is still bleeding fresh sap from the newly acquired wound. while the sap is dripping down and collecting on the ground. This MSAP stands for MSAFP, or MSAP, which really means maternal serum alpha-fetoprotein. And the fact that the MSAP is collecting on the ground represents that it's low. So MSAP on the ground stands for low maternal serum alpha-fetoprotein, or MSAP. This poor family can't catch a break. Look at this burglar stealing their HD TV. He is crawling out of the window to escape on the roof. HD TV sounds a lot like beta HCG, and the fact that the TV is exiting from a high window indicates that beta HCG is elevated. So in maternal serum, if the beta HCG is elevated, this could be an indication that the fetus has Down syndrome. So HDTV high on the house for beta HCG. I'd say the worst offense of all is the abduction of their teenage daughter. Awful. She has the letter A worn prominently on her shirt. You can see that rope wrapped around her really tight. This is inhibiting her from escaping. This inhibition represents inhibin, specifically inhibin A, as indicated by her awesome school shirt. Not sure what school she goes to, but it starts with the letter A. Now, being pulled up on the roof indicates that inhibin A is elevated. So the inhibited girl with the A shirt being pulled up onto the roof represents elevated inhibin A in maternal serum. And with that, you have learned all the items you need to memorize for Down syndrome.
Okay. So, Dr. Jordan, did you take the picture of this one? Dr. Jordan, were you able to take a picture of this one? Okay, thank you so much. So Down syndrome, did you guys understand the cartoon of Down syndrome? Everything, more or less? Don't worry about this because I know there's a lot of information over here. We'll discuss about this right now. But have you understood everything more or less? And if we talk about this, I'm pretty sure you guys can reproduce this. So let's talk about Down syndrome. Is everyone ready to talk about Down syndrome? Okay. Down syndrome. First of all, Down syndrome, it is also known as trisomy 21, meaning that there's an extra chromosome at chromosome number 21. Okay. So first of all, if I want to talk about the findings of Down syndrome, let's talk about the findings of Down syndrome. What are the findings of Down syndrome? Let's see if you guys remember the findings of Down syndrome. What are the findings of Down syndrome, please, from the cartoon which you just saw? The cartoons. What are the findings of the Down syndrome? Okay, from head, single palmar crease, then what else? Single palmar crease, epicanthic folds. Okay, 1 second. So we have single palmar crease, then epicanthic folds, then increased gap between first and second toe. Then what else? Let's see. Duodenal atresia. Then what else did you guys? Brush fill spots in the iris. Okay, then what else? High inhibin. All AML. Okay. Brush field spots in the iris. Okay, then what else? Then inhibin is high. Then you have AML and ALL. High risk of Alzheimer's disease. Very good. Alzheimer's disease. Then Hirschsprung's disease. Okay. What type of cardiovascular defect do you find in Down syndrome? What type of cardiovascular defect do you find in Down syndrome? ASD. Atrioventricular septal defect, ASD, VSD. Okay, both of them are common. ASD is also common, so ASD, VSD. Okay, good. Okay. Okay, now next one is what happens to the serum markers, the hormones? What happens to the serum markers? HCG, what happens to HCG? HCG, what happens to HCG? Increased HCG. Then what happens to inhibin? What happens to inhibin? Inhibin is high. What happens to AFP? AFP. What happens to estrogen? Okay. Do you guys realize how easy it is for you to recall all of these informations after watching the video? Yes? Okay. Okay. Now, what are the causes of Down syndrome? Then the number one cause is meiotic non-disjunction. Then we have Robertsonian translocation and mosaicism. Do you remember we talked about this? Robertsonian translocation, meiotic non-disjunction, and mosaicism. Okay. Okay. Now let's go to our text. Let's see what our text says. So we're talking about Down syndrome. In Down syndrome, let's see. Okay. Okay, 1 second. One second. Okay. Findings. We have intellectual disability, flat faces, prominent epicanthic folds, single palmar crease, increased fifth finger, incurved fifth finger, gap between first and second toes, duodenal atresia, Hirschsprung disease, congenital heart disease, ASD, brushfield spots in the iris. It is associated with early onset of Alzheimer's, which we talked about this, because you have the amyloid APP protein, amyloid precursor protein on chromosome number 21. Do you remember we talked about this in neurology? That APP is on chromosome number 21, and trisomy 21 is associated with increased amyloid precursor protein. Then we have increased risk of AML and ALL. Then 95% is due to non-disjunction, because with increased maternal age is one of the cause. And over here, it's also between in 1 to 1:1,500. That is, out of 1,500 women, one woman above the age of 20, if they have a baby, they will have Down syndrome, and 1 out of 25 women above the age of 45 years will have a baby with Down syndrome. Then we have 4% due to unbalanced Robertsonian translocation. Most commonly, the translocation is between the translocation of gene between chromosome number 14 and 21. What type of lab procedure can we do to identify this Robertsonian translocation? What type of lab procedure can we do to identify this Robertsonian translocation? FISH. Very good. We can do FISH. Only 1% of cases are due to post-fertilization, mitotic error. Also, mosaicism. Incidence is 1 out of 1,700. The Down syndrome is chromosome 21, most common chromosomal disorder, and most common cause of genetic intellectual disability. This is a high-yield question. What is the most common cause of genetic intellectual disability? And the cause is Down syndrome. In first trimester, the ultrasound will show increased nuchal translucency and hypoplastic nasal bone. The markers of Down syndrome are high up, meaning increased HCG, increased inhibin, and we also saw from physio that we have decreased AFP and decreased estrogen. So write it down, write this down in your book. Write this down in your book. Okay, that's that. These are the 5 A's of Down syndrome: advanced maternal age, duodenal atresia, atrioventricular septal defect, Alzheimer's disease, and AML and ALL. Are we clear about this? Are we clear about Down syndrome? Okay. Very well. Okay. Are you guys tired, or can we do Edwards and Patel syndrome? Can we do Edwards or Patel syndrome? Okay. All you have to do is look at the cartoon. Okay, that's all you have to do. Can you please have just a little bit more patience? We would be done with these topics very quickly. Have a little bit more patience. All you have to do is look at the cartoons and we'll be done. If we can finish these portions of biochemistry, can we skip out on doing the UWorld questions today so that we can finish the biochemistry up to nutrition? Okay, so that way we will finish on time, and we can do the questions tomorrow. Okay, we will do more questions tomorrow, but let's get done with these portions. Okay. So thank you so much for your patience and thank you so much for your cooperation and understanding. So let's begin with Edwards syndrome. Let's watch the video on Edwards syndrome. No, once again, Dr. Jordan will be in charge of taking the picture and posting it in the group. And after that, we will obviously discuss this and get done with it. It's not a long video, it's a shorter video. 4 minutes. Okay, it's just a 4-minute video. That's what we're going to do. We're going to watch the video and then
Welcome to section 10.
Okay. Okay.
Of genetics. In this section, we will be memorizing the details of Edwards syndrome. So let's get started. Our story of Edwards syndrome takes place outside of Old Man Edwards' shop. Edwards' shop for Edwards syndrome. It's actually election day, and Edwards' shop hosts one of the city's voting booths. As you can see on this window, you must be 18 years old to vote, which makes sense. We all know that you need to be 18 years old to vote. Now, the number 18 stands for chromosome 18, which is the problem in this syndrome, because there are 3 copies of it. Therefore, Edwards syndrome is also called trisomy 18. So again, must be 18 to vote for trisomy 18. As you can see, there is a closed sign outside the voting booth. This woman here has missed her opportunity to vote. That's gotta be super frustrating. You can see she is frustrated because she is clenching her fists in anger and disappointment. Her clenched fist anger is totally understandable. How could the polls be closed? It's still sunny outside. In any case, her clenched fists will help you remember the clenched fists that Edwards syndrome infants often have. Now look at those impressive gauges hanging in her ears, pulling them lower on her head. This lowering of her ears represents the low-set ears that Edwards syndrome patients are known to have. So big pulling ear gauges for low-set ears. Now look at her head high in this awesome bun. This bun kind of makes her occiput, this area of her skull, appear more prominent. The jewels don't represent anything themselves. They are simply a nice way to draw attention to her prominent occiput. Anyways, this prominent bejeweled bun represents the prominent occiput seen in Edwards syndrome patients. So prominent bun for prominent occiput. Now let's bring in Old Man Edward himself. He sits outside his shop to ensure no one can vote if they are late. Look at him point that finger of scorn at our young eligible voter. You can almost hear him say, "You're late, you whippersnapper." Anyways, look at that rocking chair he sits on. The rocking chair represents the rocker bottom feet seen in patients with trisomy 18. So rocking chair for rocker bottom feet. Now he has a trusty little dog chewing on an animal jawbone that he himself is much too small for. This jaw that is being gnawed on by the small dog represents micrognathia in these patients, or small jaw. So micro dog gnawing on a jaw stands for micrognathia. Now look at this kid on the sidewalk back here. He is trying to fix his broken car. At Physio, we like to use cars to help draw attention to cardiac issues. So a broken car represents cardiac disease. The fact that a child is here working on this broken car further emphasizes the fact that the cardiac disease is congenital. So again, a child working on broken car for congenital cardiac disease in Edwards syndrome. Now, little man back here is trying to impress the girl next door by offering her a sweet ride in his car. And I mean sweet. Look at those sweet cookies he gave her. Unfortunately, the cookies are stuck together, conjoined if you will. This represents the non-disjunction and meiosis that leads to trisomy 18. So conjoined cookies for non-disjunction. The maternal labs are often super hard to remember. So let's go over them right now. Look at the delivery truck in the background. It's driven by a pretty careless delivery driver. He forgot to close the back door, so a brand new HD TV fell out of the back. HD TV sounds a lot like beta HCG. The fact that it landed on the ground indicates that beta HCG is low. So HDTV landing on the ground for low beta HCG. As if a destroyed HDTV wasn't bad enough, the TV wrecked a nearby street sign on its way down to the ground. That poor delivery driver is racking up some costly mistakes. Anyways, the fallen sign reads E Street. E Street kind of sounds like estrogen. The fact that it has fallen will help you remember that estrogen is low. So fallen E Street sign stands for low estrogen. Now look at the tree in the background here. Notice how someone has carved their first initial into the tree. From the carved M, there is sap dripping to the ground. poor trees losing all that sap. Anyways, this MSAP represents MSAF, or rather, maternal serum alpha-fetoprotein. You will sometimes hear or simply read AFP, but often you will come across MS-AFP for maternal serum AFP. But looking at the letters and sounding it out, it sounds a lot like MSAP. The fact that MSAP is dripping down to the ground will help you remember that MSAP is low in Edwards syndrome. So MSAP on the ground for low maternal serum alpha-fetoprotein, or MSAP. Another lab you will be tested on in trisomies is Inhibin A. We didn't include anything in Edwards syndrome because Inhibin A is normal. So when you think of Edwards syndrome from this image, you won't remember anything about Inhibin A, and that's because it's normal. And with that, you have learned all the items that you need to memorize for Edwards syndrome.
Okay, good. Dr. Jordan, were you able to take the pictures?
Yes.
Dr. Jordan, were you able to take the pictures? Okay, thank you so much. Okay, very good. Okay, so let's see if you guys have learned the informations from the cartoon, which we just saw. First of all, Edwards syndrome. Edwards syndrome is trisomy 18 or trisomy 13. Which one is Edwards syndrome? Trisomy 18 or trisomy 13? 18. Okay, good. What are the clinical features of Edwards syndrome? What are the clinical features? Let's do this. Clenched fist, then prominent occiput, then rocker bottom feet, then what else? Low set ears, very good. Low set ears. Then what else? Micrognathia. Okay, then what else? Micrognathia. Anything else? Congenital heart disease, very good. Congenital heart disease. Okay. Good, very good. Okay, let me see if you have missed out on anything. Prominent occiput, rocker bottom feet, intellectual disability, non-disjunction is the cause, meiotic non-disjunction. Clenched fist with overlapping fingers like this one. Low set ears, micrognathia. Where do you find micrognathia and glossoptosis? What is the name of the disease of micrognathia and glossoptosis together? Micrognathia and glossoptosis. What is the name of the disease? Anyone? Glossoptosis, micrognathia. Where do we see glossoptosis and micrognathia? Anyone? Anyone? Nope. Robin, there we go. Pierre-Robin sequence. Do you guys remember Pierre-Robin sequence? Pierre-Robin sequence. What is Pierre-Robin sequence? What is Pierre-Robin sequence? Abnormality in the pharyngeal arch 1 and 2, first and second pharyngeal arch, abnormality. Abnormality, okay, good. Then we have congenital heart disease, omphalocele, myelomeningocele, okay? Okay. If we are sure about Edwards syndrome, can we start with Patau syndrome? It won't take that long. Yes? Okay. Okay, so Patau syndrome. Okay, is everyone ready? Is Dr. Jordan ready? Everyone? Is everyone ready? Is Dr. Jordan ready?
Yes, I'm ready.
Okay, perfect. Thank you so much. So let's do this.
Section 10.3 of genetics. In this section, we will be memorizing the details of Patau syndrome. Let's get started. Our story starts with a ground level view of a little skirmish between some toys. So let's introduce the first few toys. On the right, you'll see the hero of the story. He's our Buzzy character, and you can see him punching this toy villain. Notice that when our Buzzy character punches the villain, there's this cartoony POW symbol. This POW will help you remember Patau syndrome. POW, Patau. Notice that this punch really messed up the face of this villain toy. The punch gave him a cleft palate. This punch-induced cleft will help you remember that Patau syndrome patients often have cleft lips or cleft palates. Now look at the hand on this villain. He has 6 fingers. This signifies polydactyly, or extra fingers, which is common in Patau syndrome. So 6 fingers, polydactyly. Now our Buzzy character doesn't look that perfect either. In fact, the villain has been experimenting on other toys, swapping out parts for other parts. Our hero recently had his head swapped out for a much smaller Lego head. This micro Lego head will help you remember microcephaly, which is common in Patau syndrome. Now it's fitting that our Buzzy character has a toy halo attached to his suit. After all, he is the hero of our story. Anyways, the halo will help you remember that Patau syndrome patients often have holoprosencephaly. In holoprosencephaly, the forebrain fails to separate into 2 hemispheres. So halo, holoprosencephaly. Now our villain is a lover of all things dark and creepy, including Friday the 13th. He has taken a human's calendar and circled this ominous date. The fact that 13 is circled will help you remember that there is an extra chromosome 13 in Patau syndrome. In fact, Patau syndrome is also called trisomy 13. So Friday the 13th, trisomy 13. Remember how our hero got his head swapped out for a smaller Lego head? Well, he wasn't the only victim of this villain toy. Here is a potato toy strapped down and being tortured by a magnifying glass. See how the heat is burning a hole in his outermost layer of skin? Or at least his outermost layer of plastic. Well, this looks like cutis aplasia. Cutis aplasia indicates that a portion of skin, or even bone sometimes, is missing. If you think of a hole of skin missing, like in our potato character, you can remember cutis aplasia in Patau syndrome. Here are 2 other victims who sympathize with the burning potato. Look at this cowgirl reach out with compassion. Unfortunately, she is sewn up to the cowboy. This must have happened recently because the villain was still holding the needle and thread in his 6-fingered hand right before he got punched and received his cleft palate. Anyways, the fact that these 2 toys are sewn up represents the meiotic non-disjunction of chromosomes that leads to an extra chromosome 13. So conjoined toys for non-disjunction. Our cowboy character is dealing with his own issues at the moment. Notice how the villain plucked his eyes out. This poor cowboy can't see. He just has these tiny little stitchings now where his eyes were. These tiny stitchings will help you remember microphthalmia, or small eyes, which is common in Patau syndrome. Now the human in the background is obviously unaware of the toy-themed violence occurring at her feet. She could be overcome with relief since her medical labs recently came back normal. This medical report indicating normal labs will help you remember that mothers with a fetus affected with Patau syndrome will have a normal beta-HCG, AFP, inhibin A, and estriol. In the first trimester, the beta-HCG can be low. However, it's best to remember these labs in the second trimester and just recall that they are all normal. So normal labs with Patau syndrome. Now this careless human likes to keep a big bag of kidney beans around. She doesn't take great care of them or do anything with them. So they got all these nasty growths on them. These nasty little growths on the kidney beans kind of look like little cysts. So cysts on kidneys. So these cystic kidneys will help you remember that patients with Patau syndrome often have polycystic kidney disease. Lastly, notice how the human is on a rocking chair. You notice the interesting shape of her feet? So the shape of her feet and the fact that she's on a rocking chair will help you remember that patients with Patau syndrome have rocker bottom feet, or feet shaped like rocking chairs. And with that, we have completed all the details of Patau syndrome.
Okay, so that's that. Dr. Jordan, could you take a picture of this one? Okay, good, very good. Thank you so much. So let's do this. Let's see if you guys have learned everything about Patau syndrome. So basically, Patau syndrome is a trisomy of chromosome number 13. Yes, so rocker bottom feet is more or less common for both. Okay, okay. So over here, trisomy 13 is known as Patau syndrome. Let's talk about the clinical features. What are the clinical features which we just saw over here? What are the clinical features which we just saw? Clinical features. What are the clinical features which we just saw over here? Guys, clinical features. What are the clinical features which we just saw over here? Okay, good.
Palate.
Okay. Cleft palate. Cleft palate. cleft lip. Then we have polycystic. We have polydactyly. My apologies. Polydactyly, right? Then what else?
Microcephaly.
Microcephaly,
And the halo for holoprosencephaly.
holoprosencephaly. Then we also have congenital heart diseases. Okay, congenital heart disease. Congenital heart disease. Okay. Okay, so do we have any issues regarding remembering these findings after we have seen the pictures in physio? Yes or no? Okay, good. Okay, now let's talk about let's talk about the hormones. Okay, let's talk about the hormones. Over here, we have beta HCG. We have inhibin A. We have estrogen and alpha fetoprotein. Okay, we're talking about the second trimester findings. Second trimester for trisomy 21, trisomy 18, and trisomy 13. The second trimester findings, which are more high yield. Okay, so for trisomy 21 at the second trimester, what do we find with beta HCG? Fast answers, please. Increased or decreased? Increased. What happens to inhibin? Increased. And estrogen and AFP? Decreased. Very good. Trisomy 18, what happens to beta HCG? Decreased. Inhibin? Decreased or normal? They could be decreased or normal. Okay. Estrogen and AFP? Estrogen and AFP? Decreased. Trisomy 13, what happens in the second trimester? Which we just saw over there? Normal. Okay. What happens to the beta HCG in first trimester of trisomy 13? Do you remember they mentioned that it's decreased in first trimester, but it's normal in second trimester? Okay. So in first trimester, beta HCG is decreased. And in the second trimester, it's normal. Okay. Second trimester is normal. Along with this, we also have another thing called PAPPA. What is PAPPA? What is PAPPA? Does anyone know the full form of PAPPA? Anyone? Anyone knows the full form of PAPPA? Pregnancy associated polypeptide A. Are we clear about this? Pregnancy associated polypeptide A. Okay. This protein over here is a non-specific protein, which is decreased in all trisomies. It is decreased in all trisomies in the first trimester. Are we clear about this? Okay. Are we clear about this? Okay. Okay, good. Good. So that's that. So Patel syndrome, the findings are intellectual disability, rocker bottom feet, microophthalmia, microcephaly, cleft lip, cleft palate, holoprosencephaly, aplasia, congenital heart disease, polycystic kidney disease, and that usually occurs by age 1. First trimester screening, beta HCG for 21 is high, for these are low, and we just talked about this in this one over here. And over here, Patel syndrome, there's also another high yield thing which they mentioned is there's a defect in the fusion of precordial mesoderm. To be honest, it's not very high yield, but as a result, patients of Patel syndrome, they have the defect in the midline. So if you ever receive a question, I'm not sure if you will or not, but if you do, then remember that in Patel syndrome, there is a defect in precordial mesoderm. It's not very high yield. So if you cannot remember this, it's not a big issue. It's not a big problem. Okay. Now, what else do we have? Okay, good. Let's talk about non-disjunction. Non-disjunction is basically failure of the chromosomes to separate. You can have the disjunctions in meiosis 1. You can have the disjunctions in meiosis 2. If it happens in meiosis 1, then obviously everything is shifted on this one, this side. So this side is you have trisomies, and then you have the monosomies in equal amounts. If you have the disjunctions occurring in meiosis 2, then you can have trisomies and monosomies and normal. So if I ask you a question, at which non-disjunction is trisomies more common? Are trisomies more common at meiosis 1 or at meiosis 2? What should be your answer? Meiosis 1. And if I ask you at which meiosis is non-disjunction happening for trisomies to take place? Meiotic non-disjunction, at which phases is non-disjunction happening for trisomies to take place? The answer is both meiosis 1 and meiosis 2. But in meiosis 1, the trisomies, there's a higher chance of having a trisomy if it's a meiotic 1 non-disjunction. Are we clear about this? Okay, are we clear about this? Higher chance of having trisomies if it's a meiotic 1 non-disjunction than having the trisomies in a meiotic 2 non-disjunction. Okay, and the meiotic non-disjunction can happen in both 1 and 2. Okay, are we clear? Okay, good. Okay, before we talk about Robertsonian translocation and genetic disorders by chromosomes, I want to get rid of talking about these two conditions, Cri-du-Chat syndrome and Williams syndrome. So the best way to do this is look at the videos once again. Is everyone ready to talk about Cri-du-Chat syndrome and Williams syndrome? Okay, are you guys having some amount of fun? Okay, do we have any student over here with little babies? This is the best video. I mean, this is the best day for you to enjoy the lectures with your little babies. Make them watch the cartoons with you. Okay, call them and tell them that I'm watching a cartoon. Come and watch it with me. Okay, good, very good. Okay, so let's do this. Have you guys seen Toy Story? I'm sure you guys have, right? They use the Toy Stories over here. Okay. We are going to talk about Cri-du-Chat syndrome. Okay. Everyone with their babies, get ready.
Welcome to section 12.1 of
Dr. Jordan, are you ready? Okay, good. Let's do this.
genetics. In this section, we'll be memorizing the details of Cri-du-Chat syndrome. Let's get started. This scene takes place inside of a bank and is a classic story about a hero and a villain. In this story, the hero is Cat Lady. Cat Lady caught wind of a bank robbery, so she came to the rescue. In this image, Cat Lady is our symbol for Cri-du-Chat syndrome. In French, chat means cat, so we thought a Cat Lady superhero would go well with this idea. So again, Cat Lady for Cri-du-Chat syndrome. The villain is Penguin Man. Notice that he likes to bring along penguins on his bank robbery adventures. Unfortunately for him, his evil tyranny is coming to an end because Cat Lady is about to dismantle him. Look at how she's clawing off his arm with her claws. Those must be pretty sharp claws. This part of the scene is to help you remember that Cri-du-Chat syndrome is caused by a deletion on the short arm of chromosome 5. The fact that Penguin Man's arm is getting cut off with 5 fingers should help you remember chromosome 5. So arm getting clawed off with 5 fingers for short arm of chromosome 5. As Penguin Man's arm is falling to the ground, he screams in pain, just as anyone would do in this situation. The scream here is to help you remember that one of the clinical features of infants with Cri-du-Chat syndrome is a high-pitched meowing or screaming sound. So Penguin Man screaming for high-pitched crying or meowing. We chose Penguin Man as the villain for several reasons, but one of them is the fact that penguins tend to have very small heads in relation to their bodies. The small head here is to help you remember that one of the clinical features of Cri-du-Chat syndrome is microcephaly. So small-headed penguin for microcephaly. Next, notice that Cat Lady is destroying some sort of canister on the ground. This canister was full of poison that Penguin Man was planning on administering throughout the city's water supply. Luckily, Cat Lady smashed the canister and is taking care of the evil villain, so the city should be safe now. Canister sounds kind of like epicanthal, so we've used it here to help you remember that epicanthal folds may be seen in Cri-du-Chat syndrome. So canisters for epicanthal folds. This bank employee was minding his business when Penguin Man decided to barge into the bank and make everyone hostages. Most of the hostages have fled from the scene, but this guy isn't too bright, as you can probably tell by the way he's reading the newspaper. Notice how it's upside down? Yep, that's because this man has intellectual disability. One of the clinical features of Prader-Willi syndrome is intellectual disability. So again, upside down newspaper for intellectual disability. Okay, let's turn our attention to these dead-looking trees. Why would the bank have these ugly, dead-looking trees in their lobby? As you can see, they're being killed by this poisonous smoke coming through the vent. They weren't dead until just a few seconds ago, as Penguin Man desperately tried to kill everyone in the bank as his last act of terror. Fortunately for everyone in the bank, Cat Lady has it under control because she just destroyed the vent with her whip. The vent being destroyed here is our symbol for ventricular septal defect. Vent sounds like ventricle, and the fact that the vent is broken in a vertical direction, just like the orientation of a ventricular septal defect, should help you remember that a ventricular septal defect is a clinical feature of Prader-Willi syndrome. And with that, you should have everything you need to memorize about Prader-Willi syndrome.
Okay, so that's all about Prader-Willi syndrome. Have you guys, Dr. Jordan, have you taken a picture of the Prader-Willi syndrome? Okay, good. So let's discuss about the Prader-Willi syndrome. Prader-Willi syndrome. Prader-Willi stands for the letters cry of the cat. Cry of the cat, basically, if you can watch a YouTube video of Prader-Willi syndrome on the patient, you will see that the patients, when they cry, it sounds like they have a very high-pitched crying, almost like when a cat is crying or meowing as a result of that. That's that. So my first question to you guys is, what is the chromosomal defect for Prader-Willi? What is the chromosomal defect for Prader-Willi? On which chromosome? On which chromosome? Prader-Willi syndrome.
5.
Okay. Chromosome 5. Chromosome number 5, short arm of chromosome number 5. Okay, next one. Next one is what are the clinical features and findings of Prader-Willi syndrome? What are the clinical features?
Microcephaly.
Okay, high-pitched crying, then microcephaly, then VSD, intellectual disability, epicanthic folds. Okay, more or less, these are the ones. Okay, VSD is there. Okay, so if you guys receive a question with these type of findings, can you identify that you are dealing with Prader-Willi syndrome? Yes, okay. Prader-Willi syndrome is cry of the cat. It's a congenital deletion of the short arm of chromosome number 5. The findings are microcephaly, moderate intellectual disability, high-pitched crying, epicanthic folds, and cardiac abnormalities. The most important question you will find from over here is that the questions will come from the finding, and the diagnosis is the answer. Okay, the diagnosis is the answer over here. Okay, okay. Are we ready to begin Williams syndrome? Last one more. After that, we're done. Okay. One last cartoon. Everyone with their baby, get ready. Dr. Jordan, get ready. Okay, let's do this. Have you guys watched the movie Elf
Welcome to section.
by Will Ferrell? Do we have anyone who has watched the movie Elf by Will Ferrell? Anyone? Okay. Okay, good. Then you guys are going to love this. Yes.
Section 12.2. Welcome to section 12.2 of genetics. In this section, we'll be discussing Williams syndrome. Let's get started. This scene will take place in Santa's workshop. Notice that one of his most famous helpers, Will Ferrell, is here raising his hand very excitedly. In this scene, Will Ferrell represents Williams syndrome. Next, notice that he's holding up his arm with a list that says 7 deadly sins. Compared to this little elf guy next to him, his arm is quite long. He's holding up the list to remind Santa that kids who have been naughty will not be getting presents this year. The long arm and the 7 deadly sins list should help you remember that Williams syndrome is caused by a microdeletion on the long arm of chromosome 7. Just like in elementary school, Will Ferrell has found some yummy gum stuck to the bottom of his chair. He's pulling it out from underneath the chair in excitement because who doesn't love some old gum stuck to the bottom of a chair? The stretchy gum here should help you remember that the deleted region on chromosome 7 includes the elastin gene. So stretchy gum for the elastin gene. Also, pay close attention to Will Ferrell's face. Notice how he appears a bit overly excited considering the situation. This facial expression here should help you remember that patients with Williams syndrome have well-developed verbal skills and can be overly friendly with strangers. Okay, now let's turn our attention to this little elf guy sitting next to Will Ferrell. The first and most obvious thing to know about him is that he's an elf. This should help you remember another clinical feature of Williams syndrome, which is a distinct facial appearance, so elfin facies. Also notice that he's diligently working, preparing all of the toys that Santa will need to deliver for the Christmas season. He's currently building a toy car. The car here is to help you remember that this disorder results in cardiovascular problems. In this scene, we're going to take it one step further and help you memorize which cardiovascular problems are most common in this disorder. Notice the broken exhaust pipe that the elf is attempting to glue onto the car. This type of an exhaust pipe is a truck accessory that is usually located above the truck. So in this image, it represents supravalvular aortic stenosis. The supravalvular area is located above the heart in the aorta, just like how this exhaust pipe is located above the vehicle. So remember, exhaust pipe for supravalvular aortic stenosis. Okay, now let's add Santa to the image. In this scene, Santa is teaching the elves about kidney bean plants. Also notice that he's drawing a line right through the stem that's going to the kidney bean. The kidney bean represents the kidney organs, and the stem represents the renal artery. So this part of the image is here to help you remember that another cardiovascular problem in this disorder is renal artery stenosis. Now we've added this adorable little dog to the scene. Notice that it's wearing a shirt that says vitamin D, and that it's chewing on a bone with little crystals that are falling to the floor. This is a reference to our vitamin D video and is here to help you remember that patients with Williams syndrome have hypercalcemia due to increased sensitivity to vitamin D. This will be our last character added to the image. Notice anything odd about the way she's holding that book? Let's zoom up so you can see a little better. That's right, she's holding it upside down. She must not be thinking too clearly at the moment. The upside down book here is our symbol for intellectual disability, which is another clinical feature of Williams syndrome. So upside down book for intellectual disability. And with that, I think it's time for me to take a break and catch up on some Christmas movies.
Okay, all right, great. So that was the last one. Dr. Jordan, were you able to take the pictures for Williams syndrome? Okay, good. Okay, so let's talk about Williams syndrome over here. Williams syndrome is a genetic disease. Okay, where is the microdeletion? Is there a microdeletion or no? Okay, on the long arm of chromosome number 7 for the 7 deadly sins and the long arms of Will Ferrell. So long arm. Okay. Tree-to-chat syndrome. What is the chromosome number that is damaged? Okay. Duchenne muscular dystrophy. Which chromosome is affected? Duchenne muscular dystrophy. Oh, my apologies. I'm sorry. Prader-Willi syndrome and Angelman syndrome. Which chromosome number is affected? Prader-Willi syndrome and Angelman syndrome. 15. My apologies. Okay. Okay, good. Okay, so Williams syndrome is chromosome number 7 and long arm. Sometimes I like to mix and match some of the questions to see if you guys remember, and you guys never fail to provide the right answer. Okay, so you guys always provide the right answer. Thank you so much. Okay, so chromosome number 7, long arm. And what are the clinical features of Williams syndrome? What are the clinical features of Williams syndrome? Clinical features.
Elfin face.
Elfin facies. Then what else? Elfin facies. Then what else? Then we
Long
also have,
extremities.
okay,
Intellectual disability.
intellectual disability. Okay. What else?
Cardiovascular problem.
Cardiovascular problem. Renal artery stenosis. Supravalvular aortic stenosis. Okay, very good. These are the things. Okay, if we have a patient with renal artery stenosis, what would, can these patients have hypertension?
Yes.
Okay. If we have a patient with renal artery stenosis, can these patients have abdominal bruise?
Yes.
Okay, abdominal bruise sounds in signs and findings. Okay, make sure that you guys remember this. Okay, so let's go back to our text. So over here, we're talking about Williams syndrome. It's a microdeletion of long arm of chromosome number 7. The deleted region includes elastin gene. The gene is elastin gene. Findings is elfin facies, intellectual disability, hypercalcemia is also there, well-developed verbal skills, and extreme friendliness. They're very friendly. So we talked about two diseases where the child is, one is where the child is always smiling, that is Angelman syndrome. Another one is the child is extremely friendly, that is Williams syndrome. Cardiovascular problems can consist of renal artery stenosis and supravalvular aortic stenosis. That's that. Okay, so that's what you find in Williams syndrome. Are we clear about this? Are we clear about Williams syndrome? Yes? Okay, good. Okay. Let me ask if you guys remember some of the chromosomes which we have studied today. Once again, Cre-Du-Chat syndrome. What is the chromosome number for Cre-Du-Chat syndrome? Chromosome number 5. What is the chromosome number for Williams syndrome? For Williams syndrome. Chromosome number 7. What is the chromosome number for cystic fibrosis? Good. What is the chromosome number for Huntington's disease? Huntington's disease. 4. Very good. What is the chromosome number for Down syndrome? What is the chromosome number for Edwards syndrome? What is the chromosome number for Patel syndrome? Okay. Do you guys know the name of the disease called 22q11 syndrome? What is the name of the disease which is called 22q11? DiGeorge syndrome. If it says 22q11 syndrome, what is the chromosome number affected in DiGeorge syndrome? Chromosome number 22, very good. What is the chromosome number affected in Prader-Willi and Angelman syndrome? 15. Do you guys remember we talked about hemochromatosis? Hemochromatosis, right? Hemochromatosis. What is the chromosome number? Okay. Do you guys remember we talked about 13's disease? 13's disease. Which one is 13's disease? Wilson disease. Very good. Wilson disease. Okay, Wilson disease. And that's that. Okay. So with that being said, we discussed almost all the high yield diseases from over here. How could you guys possibly answer more or less everything from over here? Did you guys read this before? Did you guys read this before? Yes or no? No. Okay. But how could you answer them? That's very impressive. So good job. So can I give you guys a very small homework for today? Very small homework for today. Okay. The homework which I want to give you guys for today is we have discussed the high yield diseases for the chromosome numbers. Can you guys still go through this image, through this picture, and we can discuss the rest tomorrow, von Hippel-Lindau and renal cell carcinomas and this and that, which we have not discussed because those are not very high yield. We asked you the high yield ones and the rest go through them. I will ask you questions tomorrow during the revision period. Okay, during the revision period. So this is homework. I'm just gonna write it down over here so that you guys go through this. It will take at least 5 minutes or something. Okay, it will take 5 minutes. Just go through this. I'll ask you guys questions. Next one. The last topic that we want to discuss before we end our lecture is Robertsonian translocation. Robertsonian translocation is a chromosomal translocation that commonly involves chromosome pairs, that is 21, 22, 13, 14, and 15. So if you write 1, 2, 3, 4, and 5, all you have to do is add the numbers 2, 2, 1, 1, 1. And that's how you can identify all the chromosomes with Robertsonian translocation. Are we clear about this? How to identify the Robertsonian translocation chromosomes? 1, 2, 3, 4, 5, and then we add the numbers on top of that. This is one of the most common types of translocation, occurs when the long arms of two acrocentric chromosomes. Acrocentric chromosomes, what does that mean? If this is a chromosome with a central centromere, okay, acrocentric, meaning that the centromeres near the ends, near their ends. So what happens is you will have some chromosomes that look like this, okay? Some chromosomes which looks like this, but this is more common. This is an acrocentric chromosome. So this is when the chromosomes with centromere near their ends, these are acrocentric chromosomes, fuse at the centromere and the two short arms are lost. There's a high possibility for these two short arms being lost. So that's that. Balanced translocation, they do not cause any abnormal phenotype, and unbalanced translocation can result in miscarriage, stillbirth, and chromosomal imbalances. The only high yield question from over here is this one. What are the chromosomes that can undergo Robertsonian translocation? And it's these ones, 1, 2, 3, 4, 5, and 2, 2, 1, 1, 1. Okay, if you can identify this, you can identify almost all the questions with Robertsonian translocation. Another question which could be a bit high yield is acrocentric. Whenever they say acrocentric chromosomal translocation, think about Robertsonian translocations. Are we clear about this? Yes or no? Are we clear about this? Robertsonian translocation, everyone? Okay, good. So are we ready to begin the biochemistry nutrition? Are we ready to begin biochemistry nutrition? Tomorrow, okay. Okay. Dr. Dahlia is like, no, yes, now, okay. Okay, I apologize, Dr. Dahlia, but we'll be starting this from tomorrow, okay? So let's start this from tomorrow. I know each and every one of you are very tired as of right now, and I do not want to really force you guys, okay? I know you know, okay? I know you know too that it's a joke, okay? Good. I'm glad if you guys had some fun today, I'm glad you guys had fun, but the thing is, could you guys, I mean, is it better for us to use the physio because that way it's extremely easy to recall all the high yield informations? Yes or no? Yes. Okay, good. So with that being said, thank you so much for doing the lecture for today. As of right now, I want to take my leave, and tomorrow I want to see you guys at 9 a.m. tomorrow. Sure. Yes. If you have a question about ECFMG registration, you can unmute yourself and ask me the question.
Thank you, Dr. Haidari. My small question is like, I have to do like an appointment with notary and I have this paper about 186 application form, and they want to have like, my question is because there's written certification of identification by official. Do they mean like the official of notary.com, or do you have to go here in Germany to a local authority to let me to sign this for me?
Okay, good question. First of all, you're welcome for thanking me. And the question is Notary Cam and Form 186. So what Notary Cam will do is after you are done completing your Form 86, you will be provided with a link. Over that link, if you press on that, there are people in the United States who works in the notary. They will do an online interview where they will see if you are the person who you are. They will need a passport or an ID card, and everything will happen over the laptop online. You do not have to go anywhere. Okay.
Okay, good. So they're gonna sign this also for me with the.
Yes, they will sign it for you, and this signing will happen online.
Okay, perfect. Thank you, Dr. Haidari. Thank you so much.
You are welcome. No problem. Okay, so if that's
Doctor, can I ask one more small question about ESFMG also?
Yes, Dr. Hossam. What is your question, please?
I have done the notary cam since long time, and I have couriered them, and filled the online application.
Mhm.
They sent me an email that they received my courier,
Okay.
but they rejected my online application for first time. Then I resend again a new application, online application.
Okay.
That was 1 month ago. Since that time, I didn't receive any reply.
Okay. The best thing that you have to do is, if it's been 1 month and you sent it, have you tried calling the ECFMG people to see where you
Calling,
stand? Yeah. Did you call them?
I didn't know about such possibility.
No. Of course, you can call them anytime you want, 24/7. You can go to their website, take their number down, and call them. Call them and okay. So this is the thing about ECFMG.
Even if I'm not in the same country, I can
Yeah,
call them.
even if you're not in the same country, you can call them by using the country code of USA, which is if I'm correct, it's plus 8801. I'm not sure, to be honest. But if you can just find the country code for USA and call USA from the number to the ECFMG office in Pennsylvania. And the best thing is, people who work in ECFMG, they're extremely friendly and they're very helpful, and they're available to talk 24/7, especially for international medical graduates, because they know that the timing is a bit out of the ordinary. So people have been working 24/7. As a matter of fact, as soon as you're done with the class, go to their website, get their number, and call them, and see where you stand in form of your application. Okay?
Okay, you mean ESFMG.org, yeah?
Yes.
Okay, thank you for your advice.
You are welcome. Anything else? Any other question? Okay, if there is no other question, there we go, Dr. MS. Thank you so much for providing us with the ECFMG number. Dr. Hussam, this is the number. Please try calling this number and hopefully someone will help you out. Okay, so if that's all, then I would like to take my leave, and I'll see you guys tomorrow at 9 a.m. Okay, hope you guys have a great day. If there's any question, please let me know. If there's any question, please let me know, and I'll try to get back to you, or one of our team members will get back to you. Thank you, Dr. Jordan, for helping us out today with sharing the pictures. And that's that. Okay, thank you so much. I'll see you guys tomorrow at 9 a.m. Okay, please don't be late. We're going to start with nutrition, and we will start with the physio videos. All right, have a great day. Bye-bye.